हेलो, गुड इवनिंग। आवाज आ रहा है मेरा? सही से आ रहा है क्या आवाज? बताओ, कोई कमेंट में बताओ। गुड इवनिंग। रत्नेश बता रहे स्टार्ट इट क्विकली। अरे यार, रत्नेश में अभी तक पीपीटी बना के अभी अभी आ गया इधर। हाँ। अभी पीपीटी खत्म अभी किया 2 मिनट के पहले, अभी इधर आ गया ऑलरेडी। ठीक है। गुड इवनिंग, गुड इवनिंग। सदान कुमार कौर तबुटू टुबूटू। रत्नेश गुड इवनिंग। मुतेंद्र गुड इवनिंग। ओके, माय वॉइस इज क्लियर। मतलब सही से आ रहा है ना आवाज? गुड इवनिंग सर। व्हाट मेथड आर यू गोइंग टू टीच टुडे? अरे सब कुछ बता देंगे, सब कुछ बता देंगे यार। सर, इज दिस एनफ फॉर जेआरएफ एग्जाम? अरे भाई, ये तो बहुत हो गया। बट यू शुड आल्सो रीड। ओके। फाइनली द वेट इज ओवर। थैंक यू सो मच। ऐसे है तो फटाफट शेयर कर दो सबको। 100 के ऊपर जाना है, ठीक है व्यूज, उसके बाद शुरू करेंगे। शेयर कर दो फटाफट, सब सब लोग शेयर कर दो। देखो, मैं भी शेयर कर रहा हूँ सबको। आप लोग भी शेयर कर दो, शुरू करेंगे। मैं भी कुछ लोगों को शेयर करना था, शेयर कर रहा हूँ अभी। ठीक है, आप लोग भी जल्दी शेयर करो। और क्या चल रहा है? आई एम वेटिंग फॉर 2 days। गुड इवनिंग, गुड इवनिंग। थैंक यू सर, आई गॉट माय गिफ्ट। थैंक यू रतन। होप यू विल use that properly. Okay, hope it will get benefit for you. That will very helpful for me. Thank you a lot sir. Okay, देखो मेरा क्लास में मतलब और बीच में क्वेश्चन आता रहता है, ठीक है? कौन आंसर करेगा उनको गिफ्ट तो पक्का मिलेगा। देखो इधर रतन को मिल गया गिफ्ट, आप लोगों को भी मिलेगा गिफ्ट। ठीक है, बस सही से क्लास एंड तक रहना है। ठीक है। गुड इवनिंग सिंचन। दिस मैराथन लास्ट अराउंड ओ मच टाइम सर? अरे वो तो मुझे पता नहीं, मैं बस देख रहा है व्यूज व्यूज के। ठीक है, थोड़ा बढ़ा दो, सबको शेयर कर दो, इसलिए मैं शुरू कर सकते हैं। आज का पार्ट 2 होने वाला है, बहुत इंपॉर्टेंट भी है। ठीक है, आज हम ब्रीडिंग मेथड्स, सारे सेल्फ क्रॉस, एसेक्सुअल प्रोपेगेटेड, सारे क्रॉप्स का ब्रीडिंग मेथड्स और प्रिंसिपल, ठीक है, सब कुछ खत्म करेंगे आज। ठीक है, वो थोड़ा क्वांटिटेटिव जेनेटिक्स, बायोमेट्रिकल जेनेटिक्स, उसको भी टच करेंगे। ठीक है, उसके बाद सारे ब्रीडिंग मेथड्स वी विल कवर ईच एंड एवरीथिंग। आई वांट ए गिफ्ट आफ्टर 4th जुलाई। सिंचन सिंचन, ठीक है। आप सिंचन, आप मुझे बताओ, आप हमारा कोर्स में एनरोल हुआ है नहीं हुआ है? नहीं हुआ तो गिफ्ट तो आप लेना ही पड़ेगा पहले। स्टार्ट सर, आई हैव शेयर्ड द लिंक। ठीक है, 50 तो हो गया, अभी शुरू करेंगे। ठीक है, दिस इज अबाउट पार्ट 2 प्लांट बडिंग का। ठीक है, हम अभी सेल्फ इनकम्पैटिबिलिटी तक खत्म किया है। अब और अभी थोड़ा क्वांटिटेटिव जेनेटिक्स टच करके उसके बाद ब्रीडिंग मेथड्स कवर करेंगे। ठीक है, उसके पहले आई वांट टू टेल यू वन थिंग, that we have launched our 2027 course, okay, for 2027 and 2028. and also we have shared our Agri journey from how it started and what we have achieved. ठीक है, ये वो YouTube वीडियो का लिंक डिस्क्रिप्शन बॉक्स में है, ठीक है, सारे उसमें जाओ, ठीक है, कमेंट मतलब उस वीडियो देख के आपका जेन्युइन फीडबैक उसमें डाल दो, ठीक है। हमारा स्टूडेंट्स के तरफ मतलब प्लांट साइंस का स्टूडेंट्स के तरफ ज्यादा तो ज्यादा कमेंट चाहिए, ठीक है, मिनिमम आई नीड 100 कमेंट्स फ्रॉम माय साइड। इस आप प्लांट साइंस का नाम ले लो, ठीक है, उसमें आप जेन्युइन फीडबैक डाल दो, 100 के ऊपर आ रहा है, उसके बाद ही आपका पार्ट 3 मैराथन आएगा, नहीं हो तो मैं तो नहीं करेगा, ठीक है। कमेंट्स तो जरूरी चाहिए, वो वीडियो का लिंक इस वीडियो का डिस्क्रिप्शन बॉक्स में है, ठीक है, जाओ उसमें कमेंट कर दो। अभी अभी तक कोई नहीं मतलब काउंट नहीं किया, जाके अभी फटाफट कमेंट कर दो। बाहुबली पार्ट 2 अरे धुरंधर 2, ठीक है, बाहुबली तो पुराना हो गया, अभी हम धुरंधर 2 चालू करने वाले हैं, ठीक है। सर हाउ टू ओवरकम स्लीपिंग लाइक यू एट नाइट अरे मैं तो मतलब रात में करता है सारे काम सर आई ऑलरेडी सजेस्टेड माय जूनियर बिकॉज़ आई वाज लेट टू फाइंड दिस अवेजम चैनल। ओके थैंक यू, ओके थैंक यू सो मच, वी विल स्टार्ट, वी आर नॉट गोइंग टू वेस्ट टाइम नाउ। Anyway, you already knows about me. Just I will give once again my introduction. I am Abhishek. I completed my undergrad. I am from Karnataka. I completed my undergraduation from US Bangalore, and I got 21st rank in ICAR PG exam. Then I got admitted to MSc in IRI New Delhi. Then I got 4th rank in ICAR JRF or SRF exam. I also cleared ASRB Net Joint CSR UGC Net. Okay, and I am currently pursuing PhD in Division of Genetics in ICAR IRI New Delhi. I also visited Greece and Italy for one of my PhD summer school. ठीक है, ये तो मेरा brief introduction है. Today we will discuss, as I told, from starting we will give some introduction to quantitative genetics so that you will get an idea, okay, why we need to understand the genetic basis of a trait. Okay, before starting breeding methods you should know the genetic basis of the trait, how it is inheriting, okay, how it is inherited, and also the genes, okay, which are controlling it and how they are acting, okay, maybe whether they are in additive gene action, whether they are in dominance gene action. Based on that only we can choose the breeding program, right? If it is self-pollinated crop plant, which are the traits we have to target, okay, trait to matlab commonly in both the crops majorly will target yield or any the correlated traits, but the thing is, okay, how they are controlling, maybe in which gene action they are, okay, the genetic constitution of a crop plant, it is in heterozygous, homozygous, okay. To know those things, okay, you should know first whether it is qualitative trait or quantitative trait, then how it is inheriting. So that we will see first. Improving a crop that requires two things, okay. Without genetic variation, okay, as I told in the previous classes also, in our live classes also, without genetic variation, okay, plant breeding is nothing, okay. The major two things, okay, in improving any trait, you should have genetic variation, then you should go for selection. You should create, if there is no genetic variation in the base population, then you should create the variation, then you can go for selection. Then if a crop does not have variation for a trait, as I told, okay, you can create variation through hybridization, mutation, or polyploidy, any tools, okay. These are the tools, we can call them as a tools. Selection means choosing the best plants from the population, okay. It is usually based on the phenotype, but now at present we are selecting a trait based on the molecular level also, okay. Using molecular markers we are selecting a trait, but previously, okay, by just seeing the phenotype we are doing the selection. Phenotype depends on the plant genotype and environment. We will specifically we will discuss about this thing, okay. P is equal to G plus C in coming slides. You can see the genotype and phenotype. Phenotype is the visible expression of a trait, for example plant height, flower color, any trait, okay. For example your flower color, then fruit size, fruit shape. These are the phenotypes. Plant height, and you can also, there will be numerous indirect traits which are contributing for the yield. Yield is also one of the phenotype. Okay, for yield there will be a number of grains, the grain size, okay, the 1000 grain weight. These are the different indirect traits which will finally will give into the yield. Okay, it has two components. Those are your heritable and non-heritable. Okay, your P, that is your phenotype, is equal to G plus C. Your phenotype is not always controlled by genotype. Okay, in sometimes there is also influence of this particular environment. So you should know this basic understanding. Okay, P is equal to G plus C. In any condition, okay, if you are seeing the quantitative traits, okay, for example, yield, okay, you can't directly tell that, you can't directly estimate the genotypic effect and environmental effect. Okay, if it is giving some 60 quintal per hectare, okay, any variety is giving 60 quintal per hectare. Okay, you can't directly tell that out of 60 quintal, okay, 40 quintal is from genotype and 20 quintal from environment. You can't say that. Okay, for in that case you will estimate the variance, the variation present in that particular population. You can't directly tell that your your yield is due to this much, for this much genotype, for this much environment. Okay, whenever you are studying quantitative traits, you will always talk about variances. Okay, you can't directly, as I told, directly can't estimate the values. Okay, we will see what exactly the variance is. Before that, qualitative trait and quantitative trait, quantitative characters. Okay, the character is any morphological, as I told, anatomical or biochemical, any trait. Okay, your quantitative traits are controlled by one or few genes. For example, oligogenes. It is least influenced by the environment. Show distinct categories, like in case of your plant height, okay, tall and dwarf. In F2, in case of Mendel experiment, 1:2:1 in case of genotype level, 3:1 in case of phenotypic level. You can easily distinguish between the different categories. But in case of quantitative characters, controlled by many genes, okay, polygenes, strongly influenced by the environment. Due to that environment, you can't clearly distinguish between the different categories here. Okay, if for example, if you take yield per se, okay, there will be so many in the individuals in the population. Maybe they will, some will give 10 kg, some will give 20 kg, some will give 30 kg, some will give 40 kg, and in between also like 15, 25, 35. Okay, so that it shows continuous variation and measured as height, for example, yield. Okay, there will be a no clear cut categories. Your graph will come like this. Okay, if it is clear cut categories, then it's like 1:2:1. Okay, 1:2:1. But here in this case, it will merge. Your values will get merged, so that in quantitative traits, since the effect of environment and there will be more number of genes, so it is very difficult to distinguish or classify the groups. Okay, that's why it will show the continuous variation. But both follow the Mendelian inheritance. Okay, at the gene level, both will follow the Mendelian inheritance, like large and clear in qualitative traits, but gene effects are small and additive in quantitative traits. Okay, in case of qualitative traits, you can see the dominance effect also, but in case of quantitative traits, it's very difficult to, as I told, differentiate. So each and every genes will give some small and additive effects. Okay, therefore different methods are used to study qualitative and quantitative trait. If you see the differences, okay, the difference between qualitative and quantitative traits, it is controlled by one or few genes like oligogenes. In case of quantitative characters, controlled by many genes. The gene effect is large and easily detectable. Okay. In case of seed color also, pink, white, you can differentiate. Okay, but in case of quantitative traits, which are each individual gene effects are very small and additive in nature, so that you will get an continuous variation. Okay, it's very difficult to distinguish between the different groups. Show discontinuous variation, continuous variation. Trait falls into distinct classes, but no clear-cut classes. Values will overlap. Okay, that's why now you will get a continuous smooth curve. Little influenced by the environment. Again, there is a strong influence by the environment. Here you can count and or you can classify the groups. Okay, but in this case, it is due to measurement. That's why your statistics will come here. Okay, the mean values, variance. Okay, where you will apply those in case of quantitative characters. Okay, mean, variance. That's why always when you are measuring the quantitative traits, you will talk about variance. Okay, the phenotypic variance, genotypic variance. Then genotypic variance will be divided into additive, dominance. Okay, all will be estimated as variances. Okay, we are not able to directly tell that the real value of genotype. Okay, we will always talk about the variation present in the population. Okay, we can estimate that variation. We can tell that the variation is due to genotype or environment. Okay, but we can't directly tell that the genotypic effect on the trait. Okay. क्या कुछ कमेंट डाल रहा है क्या कोई? क्यूमुलेटिव इफेक्ट ऑफ इंडिविजुअल जीन्स. यस. अरे जॉनसन एक्सपेरिमेंट सारे डिस्कस करेंगे. ठीक है सर. ग्राफ नेम इन क्वांटिटेटिव कैरेक्टर बताया था ना मैं. कंटिन्यूअस वेरिएशन. ओके, विल दैट इज नॉर्मल डिस्ट्रीब्यूशन. इट इज नथिंग बट द नॉर्मल डिस्ट्रीब्यूशन कर्व. ठीक है. इट फॉलोज सिंपल मेंडेलियन रेशियोज. It does not show clear Mendelian ratios. Example, is it told flower color, seed shape, your plant height, yield, weight. Selection is easy and effective. You can easily see the flower color. You can easily see the plant height. You can easily see the seed coat color. You can choose whatever you want. But in case of here, in case of quantitative trait, for example, yield, it's very difficult to choose the plants based on the phenotype. Okay, you should go for F6, F7 generation so that plant should be homozygous condition. Then only you can able to take a chance. Okay, it's very difficult in case of quantitative traits. That's why we will measure indirect traits. Then we will see what are the traits which are influencing the particular quantitative trait. Based on that, we can go for selection. Suitable for qualitative genetic studies, and it is biometrical. As I told, quantitative genetics will be studied by biometrical genetics. Inheritance of quantitative character. It is controlled by several genes. As I told again, it is repetition again, small and cumulative effect. These are highly influenced by the environment. They show continuous variation, your normal distribution. Okay. When large number of observations are plotted, you will get a bell-shaped curve. Okay. Someone is asking, bell-shaped. The bell-shaped is nothing but normal frequency distribution curve. Okay. Because of continuous variation and environment influence, the inheritance is studied using statistical and mathematical methods. That's why there is a special branch, biometrical genetics. Biometrical genetics, sometimes we will merge with quantitative genetics, but it's not like that. Biometrical genetics will contain two branches. Okay, population genetics and quantitative genetics. In population genetics, we will target qualitative traits based on the gene and genotypic ratios in that particular branch. But in case of quantitative genetics, we will highly target quantitative traits, how they will inherit, what are the gene actions, okay, the variance, covariance, each and everything. At this point of time, you should know biometrical genetics will divide, means having two branches. One is population genetics, another one is quantitative genetics. In population genetics, we will study about qualitative traits by using gene and genotypic ratios. Okay, that is Ardy-Weinberg law. Population genetics is nothing but Ardy-Weinberg law. But in case of quantitative genetics, we will study the quantitative traits. Okay. Then, as I told, yeah, you can see here, the science that deals with the application of mathematical and statistical concepts and procedures to the study of biological problems is biometrical genetics. The branch of genetics which utilizes the various mathematics concepts and procedure for study of inheritance of a character is biometrical genetics. In any biological problems, if you use any mathematical applications, then it is biometrics. But particularly if you are using for inheritance, to study the inheritance of character, then biometrical genetics. As I told, your biometrical genetics will be two branches. One is population genetics and quantitative genetics. Okay, in population genetics, it is inheritance of qualitative characters with frequency of genes and genotypes. Nothing but your Ardy-Weinberg law. It is completely about Ardy-Weinberg law. Okay, how your genes and genotypic ratio will change. Okay, in the population when there is a random mating. Okay, in quantitative genetics, study of inheritance of quantitative character or polygenic characters. Okay, sometimes, as I told, this biometrical genetics and quantitative genetics are often used as synonyms. History, how your quantitative genetics started. अरे क्यों व्यूज तो क्यों गिर रहा है? हाँ, थोड़ा बोर हो रहा है क्या? अरे, you should know the basic concepts. Okay, before starting breeding methods, you should know the basic concepts. That's why I am telling. We will start breeding methods. Just before that, you should know the heritability. Okay, if there is a low heritability, which methods you should choose. Okay, if you are having high heritable traits, then which methods you should choose, and based on the gene actions also. That's why we will talk about these things. Then we will move to breeding methods. बोर हो रहा है क्या? बताओ कोई. समझ आ रहा है ना? मतलब, are you getting it right? ठीक है. नो सर, मतलब किस लिए नो बता रहा है? Regarding history, okay, everything is started from the Mendel. ठीक है, अभी तक क्या हुआ है, okay, सब कुछ सबके लिए बैकग्राउंड और बेसिक फाउंडेशन तो मेंडल से शुरू हुआ है. ठीक है, मेंडल and qualitative variation. Okay, everything is started from qualitative traits. In pea plants, when he selected 7 traits, all are qualitative traits. Okay, then what in that era, okay, who are following the Mendel, okay, everyone is believed that, okay, the traits are qualitative in nature. Okay, you can easily distinguish between the different categories in the ratios. Okay, Mendel studied traits which are clear-cut categories. That's why, okay, he can able to publish the two principles, the law of dominance, law of segregation, and law of independent assortment. What he gave, it's because of qualitative traits. Okay, then these traits show discontinuous variation. Individual can be easily classified. That's why qualitative traits. But okay, but there are some traits which are quantitative in nature. For example, height, yield, skin color. Okay, we are not able to differentiate, or we are not able to distinguish between different categories. Okay, individuals cannot be grouped distinctly. So there will be a era of quantitative characters started. Okay, in animal breeding, in plant breeding, it is very important. Okay, yield is important for us. Okay, anyway, to develop some principles, okay, to give a foundation for genetics, we started with qualitative traits. Okay, qualitative characters. But when the era started to begin, okay, when we applied those things to our plant breeding, okay, in our case, yield is very important. Okay, in case of yield, we can't directly distinguish the different categories. Okay, like 20 kg, 30 kg, 40 kg. Okay, it's not like that, right? It is a continuous variation. The trait itself is a continuous variation. So there will be a emerging of quantitative genetics. The first time Galton studied continuous variation, and he told it is blending inheritance. Blending is nothing but, as I told, there is no clear-cut explanation categories. Okay, Pearson applied statistical methods to biology, used mean, variance, and covariance to analyze these traits. Okay, when you are not having an option to categorize the traits as a distinguished groups like ratios or numbers, okay, there should be something to measure those traits. Okay, those are like mean, variance, and covariance. they proved continuous variation as a hereditary component. This is also the quantitative characters also having hereditary component. They will also follow the Mendelian principles. Then after discovery of Mendel, okay, the rediscovery of Mendel laws, Mendelians focused on discontinuous inheritance. Okay. Then biometricians believed in continuous or blending inheritance. So for this, you should remember, okay, Yule in 1906, he told that continuous variation can result from many genes with small effect. Okay, or continuous variation or quantitative characters is controlling by many genes with the small effects. And in Johnson in 1900, okay, when he given that pure line theory, okay, we'll discuss what exactly it is. Okay, he also told that, okay, your phenotype is due to genotype plus environment. So there is an influence of environment on the quantitative traits. Okay, difference within the pure lines are due to environment. If there are 10 pure lines, everything are same, okay, homozygous and homogeneous. If there is any variation present in that particular population, then what about that? Okay, any variation within the pure line, your genetic constitution is same, okay, everything is same, then you are getting variation. That means it is completely due to environment, no doubt about it. Okay, that's what he told, Johnson. But you will told that it is result of many genes with small effects. But it is proved by Nielsen and Haley in 1909 again. Okay, the study on seed color in wheat and oats. Okay, when he studied seed coat color, okay, there will be a different F2 ratio. If it is controlled by one gene, 3:1, controlled by two gene, 15:1. Okay, if it is 63:1, then it is controlled by three genes, as per Mendel rule. Okay, when he analyzed this detailed analysis of this particular ratio, okay, you can see here, when there are two genes which are controlling the trait, okay, yeah, he is able to differentiate the particular classes into different groups. Okay, dark red, you can see dark red. If all the alleles, positive alleles, all four positive alleles, then medium dark red, three positive alleles. Okay, three positive alleles, you can see. Then medium red, okay, medium red, only medium dark red, okay, medium red, yes. इंटरमीडिएट लिखा है इसमें, ठीक है. डार्क रेड, मीडियम रेड, इधर मीडियम डार्क रेड है ना, ये मीडियम रेड है इधर. ओके, इधर मीडियम रेड है ना, वो इंटरमीडिएट रेड है. ठीक है, बेस्ड ऑन द नंबर ऑफ अलील्स. ठीक है, यू कैन टेक इट एज डार्क रेड, मीडियम रेड, dark red, medium red, then intermediate red, then light red. ठीक है, based on the number of positive alleles. Okay, when there is a two genes which are controlling the trait, one R1 and R2. Okay, when you crossed those particular genes and when you observed the F2 ratio, okay, then they will segregate like this. You can see 1:4:6:4:1 ratio when you are considering two genes. Okay, if it is only if you are only classifying them into red and white, okay, what about the ratio? 15:1. Okay, when there are two genes controlling the traits, okay, dihybrid cross ratio, and when you are only able to classify into two groups, then you can observe this 15 are red, one is white. Okay, but when you see those into very cleanly manner, then you are able to differentiate into again the different classes. These are the classes: dark red, medium red, intermediate red, light red, and white in 1:4:6:4:1 ratio. What that means? That means each gene, okay, each quantitative traits are controlling by many number of genes, okay, with small additive effect. That's what he told in his experiment. Seed color controlled by 2 genes with additive effect. Here there is no dominance, nothing, just additive. If there are 3 alleles, okay, 4 positive alleles, then dark red. If 3 positive alleles, then medium red. Then if there are 2 positive alleles, then intermediate red. If it is 1 positive allele, light red. Then if it is there are no positive alleles, then white, completely white. Okay. Each dominant or positive alleles contributes to the redness. More positive alleles, darker the color. Genotypes with equal number of positive alleles show same intensity. What he concluded, traits can be governed by multiple genes with small cumulative effect. This is given by Yule, but experimentally, okay, experimentally proved by Nielsen and Yale. This formed the basis of multiple factor hypothesis. Who given multiple factor hypothesis? You should tell that Nielsen and Yale. Okay? 2n plus 1 for number of phenotypic classes. Yes. The more dominance gene, the darker the trait. कुछ गड़बड़ हुआ है क्या? व्यूअर्स तो नीचे जा रहा है, आगे जा रहा है, पता नहीं क्या चल रहा है. Okay. Then the second experiment, okay, yeast experiment. Study on corolla length in Nicotiana. Okay. What he did, he crossed 40 millimeter and 93 millimeter corolla length, 2 different parents. You can see here. This is corolla length, okay, 40 mm and 93 mm. He got in FN 63 mm intermediate, but F2 showed the continuous variation. Okay, this is your F2. It is continuous variation. This is F1. What he observed in this particular experiment, parents were nearly homozygous. Okay, parents are homozygous but showed slight variation. Okay, you are seeing the variation, right? This is variation. Okay, here also there is a variation. Since it is quantitative trait, okay, if you take the any parents, if you measure the corolla length, yeah, they are giving nearly 40 mm, but there is some slight variation, okay, in the corolla length. If it is, for example, if it is any qualitative trait, then you can easily select some are the red seed coat color, some are white seed coat color. But in case of quantitative, you can't do that. Okay, maybe there will be some variation. That variation is due to environment. Since it is quantitative trait, okay, in pure lines, as I told, if there is any variation in homozygous material, that is due to environment. Then F1 mean was intermediate, that supports the additive gene action. Okay, why always quantitative traits, okay, which shows additive gene action? This is the case. Okay, this is experimentally proved already. Yeah, there will be some dominance variation, but majorly there will be additive gene action. Okay, they will the small and cumulative effect. That's what you will call as additive gene action. Then variability in F1 similar to parents. Variability in F1 is similar to parents. If there is any variation, okay, variation is by variation to pata hai na, variation. This is variation. How much variation it is? Okay, it starts from, in case of 40 mm, it starts from 35 mm and 45 mm. There is some, that difference is 10. That is variation. Okay, the deviation from the mean value, okay, that is variance. समझ रहे हैं ना? Okay, you can see the deviation. Here also you can see the deviation. Here also you can see the deviation. ठीक है, मतलब कुछ तो सेम है ना, मतलब average it is very similar to parents. That is due to environmental effect. Again, if there is any variation present in F1, that is again due to variation. Then F2 showed much greater variability. You can see, this is your F2. You can see the variation in case of F2 due to segregation and recombination of multiple genes. Okay, since there are multiple genes are present, there is a segregation and recombination. That's why you are getting the more variation in case of F2. Then if you go from F2, F3 like that, you can see if you select these plants, then you will get a variation at that particular level. Okay. Again from F2 onwards, if you go for selfing, okay, the variation will be decrease. Okay, within the line. This is one line. If you take some line here and if you go for next generation, you can see this is the F2 variance, but in the next generation your variance is getting decreasing within the line. Again within the line your variance is decreasing. See, but if you take overall among the lines, okay, again it is like maintaining or increasing. बताओ समझ रहे हैं ना? Yeast experiment. This is what yeast experiment is. Okay, parents are homozygous, nearly homozygous. If any variation within the parents, that is due to environment. In F1 it is comparable to your parents. Again if any variation in the F1, then again it is due to environment. But F2 there is a lot of variation. It is due to segregation and recombination. Okay, then after that if you go for again the next generation selfing, okay, the variation within the line will decrease, but among the lines, if you consider all among the lines, then your variation is maintained or it will increase. इतना ही, ठीक है? बताओ, सब बताओ अभी, समझ गए ना? ईस्ट एक्सपेरिमेंट सिंपल, इतना तो पता होना चाहिए, ठीक है? If you know this, then you are eligible to do masters in genetics and plant breeding. Okay. Then the term polygene, okay, given by Mather in 1943. Okay, after multiple factor hypothesis, okay, what your multiple factor will tell? It will tell that small and additive gene effect. But what the polygenic system will tell? Okay, you can see here. Genes with small individual effects work together and interact with other genes. Okay, interaction, epistatis. Okay, different gene combination can produce same phenotype. Often show pleiotropy, modifier or suppressor effects, formation of balanced gene systems. Okay, now we have accepted this system. Okay, polygenic system. Your multiple factor hypothesis will tells that, okay, the genes which are controlling quantitative traits are small individual effect which are additive in nature. But as I told, it's not always like that. There will be a interaction among the number of genes. Okay, so now we have accepted this polygenic system which is given by Mather. The genes which are responsible for quantitative traits are called as polygenes. But again, now the modern definition, now we are measuring it as a recognized as a QTL, quantitative trait loci. What exactly it will tell? The region which is responsible or which is controlling the quantitative traits and which contains two or more number of genes. Okay, the region which controls the quantitative traits which control more than two or more number of genes. Okay, as we are now dealing with molecular markers and everything, the sequencing. Okay, now we are given term, okay, QTL, quantitative trait loci. Multiple factor hypothesis. Then yes, there is some interaction between the genes, the presence of epistatis or dominance. Then your polygenic system given by Mather. Then at this molecular level, this era, we are calling that particular region as a QTL. Okay, quantitative trait loci, the term given by Gelderman. Okay, Gelderman. Note कर दो सारे. Next, we already now we got that. Okay, how we can measure the variance in the population. Okay, when you are talking about quantitative traits, as I told, you should always measure in terms of variance in the variation present in the population. Okay, as I told already, your variance, the phenotypic variance is equal to variance due to genotype plus variance due to environment. Okay, this is the basic definition what you should know. Now, how you will calculate this particular variance? Okay, variation. First, we will talk about variation or variance. You are having field, okay, the population. Out of that, some plants are giving 20 kg yield. Some plants are giving 30 kg yield. Okay, 20, 30, 40, anything. Okay, what about mean value? The mean will be 40, for example. And the values spread from 20 to 60. Okay, your variation or variance is nothing but the deviation from the mean value. Your mean value is 40, but the deviation you can see from your left side, from it will start from 20, from right side up to 60. The variance is around 20. That is the variation present in the population. We will always target that variation in case of quantitative traits. We will not talk about the how much yield it is giving. This line, that line. Okay. We will see the variation present in the population. Okay. That's what. That variation is due to genotype or environment. Okay. How we can calculate it? Okay. The simple experiment. You will grow. You will take some lines for 100 lines. Okay. You will grow in RCBD design, randomized block design. ठीक है, वो तो आपको पता है. There will be a blocks and there will be a replication. The genotype, okay, the variation, if any variation within the genotypes, okay, that will give you the mean square value. Okay, the error value plus replication into variance due to genotype. It is all about statistics. Okay, and your error is directly, the error will directly give the environmental variance. Okay, if you want to calculate genetic variance from this particular RCBD ANOVA, okay, you can see this is genetic variance. Variance due to genotype. The whatever variation present in the field, okay, whether it is due to genotype or environment. If it is due to genotype, then how we can calculate this value? Okay, the mean summa square due to genotype, means summa square due to genotype minus mean square due to error. Okay, divided by R. You can see, you can take the variation, the error variance, the mean square due to genotype is error variance, that is this value, error variance plus R into variance due to genotype minus MS error, that is your variance due to error again divided by R. Okay, your error will get cancelled, then your RR will get cancelled. You are end up with this particular genotypic variance. Okay, you will now we are having genotypic variance from this particular experiment. Okay, experiment is nothing but simple. You will grow genotypes, number of genotypes in RCBD design in the field. Okay, then when you will calculate those through the statistical way, okay, due to genotype, okay, whatever variance is getting, it contains error variance plus genotypic variance. Okay, then you will minus this through the error variance and you calculate it by the number of replication, you will get the variation due to genotype. This is a simple experiment. Then again, there will be so many different biometrical techniques. Okay, the diallel cross, partial diallel cross, there will be number of different crosses. By making those crosses also, you can find out, okay, the genetic variance, variance due to genotype, variance due to additive gene action, variance due to dominance gene action. That is not that much important for you guys. Okay, for you guys, the basic understanding. Okay, your phenotypic variance is controlled by genotypic variance and environment variance. Okay, how we will calculate that genotypic variance? Through RCBD design, through ANOVA. Okay, that much you should know. Please explain a little bit in Hindi. Number of replication. Yes, R stands for number of replication. अरे हिंदी में भी मिक्स कर रहा है ना, ठीक है, मैं दोनों में बैलेंस करता हूँ. ये आपको VG मिल गया, ठीक है, you got genotypic, the variance due to genotype. Then, as I told, your phenotypic variance is nothing but Variance due to genotype, variance due to environment. Anyway, you got variance due to genotype, and the variance due to environment, you will directly get it from error. Okay, you will directly get it from error. If you add both those variances, then you will end up with phenotypic variance. ठीक है? ओके, मुझे मतलब कॉन्फिडेंस नहीं आ रहा है, आपको समझ आ रहा है नहीं आ रहा है, वो बताओ पहले. What you should know is the phenotype. Anyway, as we are discussing about quantitative traits, okay, as I told, you can't directly tell that your trait, this much of a trait, the trait value is contributing from genotype, or this much amount of trait is contributing by environment. We can't directly tell that, okay, since we are talking about quantitative traits. So we should always measure the variation in the population. Okay, how we will measure that variation in the population? Through the variance. Okay, so that we can easily tell that that particular variance is due to genotype or environment. Okay, in the pure lines, everything is same. Your genotype, your genetic constitution is same. The value, each and every individual loci are same in case of pure line. Okay, but you are getting again variation. What that means? Okay, in pure lines, if you are having any variation, that is completely due to environment. Okay, that is not due to genotype. Your pure line is giving 100 quintal per hectare. Okay, that means don't get confused that 100 quintal per hectare is controlled by genotype. We are not talking about it. The variation we are talking about it. If any variation present in that pure line, then we can easily tell that it is not due to genotype. Okay, that variation is due to environment. But in case of segregating material, okay, in case of any F2, F3 material, if you are getting any variation, then we can't directly tell that it is due to environment. Okay, there will be so many factors. Your segregation will happen, recombination. So in that particular area, we can tell that it is due to genotype also. It's coming, sir. What type of question can be asked from them? As already I am telling it. Okay. The thing is, the whatever important thing you should know, that's what I am telling. Okay, you should know that you in quantitative traits, you will measure the thing in the variance. Okay, the variation you will measure. How we will measure? Through the variance. Okay, that your phenotypic variance is controlled by genetic variance due to genotype or due to environment. Okay, how you can calculate the genotypic variance? Through simple method RCBD. That much you should know. Okay. Then the Fisher in 1918 divided your genetic variance. Okay. Out of this particular genetic variance, he divided into additive variance, dominance variance, and interaction variance. That is your epistatis. Okay. This VG, then VD plus VA plus VD plus VI. Then he again divided into 3 factors. Okay, the variance due to genotype, maybe it is due to additive gene action, maybe it is due to dominant gene action. Okay, the variation which is causing. Okay, or maybe it is due to epistatic gene action. Then later epistatis was further divided into, okay, your epistatis further divided into additive into dominance, or dominance into dominance, whatever it may be. Okay, right? E classified into additive variance into and non-additive variance. Okay, additive variance and these two as non-additive variance. E classified like this. Matter E classified into irritable, fixable, non-fixable. Okay, your both additive variance, dominance variance, and interaction or epistatic variance. Okay, these are irritable in nature. Okay, our environmental variance is not irritable. It is completely due to environment. This is not irritable in nature. But your genotypic variance is irritable. Among this particular genotypic variance, he classified into fixable and non-fixable. Okay, which variance is fixable and why? Can anyone गिफ्ट मिलेगा, बताओ विच वेरिएंस आउट ऑफ दिस कैन बी फिक्सेबल एंड वाई? कोई बता सकते हैं क्या? एडिटिव 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 इज फिक्सेबल, ठीक है, सबको पता है एडिटिव इज फिक्सेबल। वाई? वाई इट इज फिक्सेबल? हाउ वी विल फिक्स, वो बताओ मुझे। एडिटिव, हाँ एडिटिव, एडिटिव ड्यू टू क्यूमुलेटिव इफेक्ट, हाँ वो तो पता है सिंचन। आई एम आस्किंग व्हाई इट इज फिक्सेबल? ड्यू टू फर्दर जनरेशन नॉट सेग्रीगेट, ठीक है, it is not segregate, but there is some reason, okay, why it is fixable in nature. It is connected to selection, okay, I will give an int, it is connected with a selection. Respond for transcriptive segregants, do not segregate, हाँ पता है, because there is a difference between homozygotes and heterozygotes. Selection, selectable, can't find out, ठीक है, I will explain, don't worry. Before that, you should know what exactly additive variance and dominance variance, okay, we will see. For example, you can consider an loci, capital A, ठीक है, and small a, and your mid parent value, okay, this is your mid parent value. Additive for particular loci, okay, additive, if it is any variation you are getting in the population, it is due to additive. Then what exactly it is? The difference between capital A and small a. The any difference, okay, if it is giving 20, then the capital A value will be 20, sorry 10, 10, then it will give 20. Here the value will be 5, 5. Here 10, 10. That's why you are getting 20, 5, 5. Then your value will be 10. Any difference due to the extreme homozygotes, okay, that variance we will call it as a additive variance, ठीक है. Your mid parent value, what about the mid parent value? 10, 20, 30, 15. 15 will be your mid parent value. But the capital A, small a is deviating. Okay, it is giving 17. The deviation from the mid parent value. Okay, anything which is causing the variation is due to this particular phenomenon is called dominance variance. The variance which is developing from this particular phenomenon, okay, that's we will call it as a dominance variance, ठीक है. Here they have took two loci, that's why they are telling for A loci difference between capital A and small a, for B loci difference between capital B and small b. You can see the dominance for A locus, deviation of AA from the average of A and A, this is average, okay, that any deviation from this particular value, that will tells you the dominance variance, okay. Like this, if the number of genes are acting this particular dominance phenomenon, and in the population you will get some variation due to this particular phenomenon, that variation we will call it as a dominance variation, ठीक है, समझ गया ना अभी, simple. Then same for dominance for B locus, okay. Then epistatis, when epistatis will come to an act? When there are two genes present, okay, additive into additive. For this particular A loci, there is an additive gene action, and for B loci also, again if I take B loci, then it is acting also additive gene action, Okay, then if there is interaction between these particular two additive into additive, then your epistatic will be additive into additive. If one is acting additive, another one is acting dominance, then additive into dominance. If both are acting as dominance, then both are dominance. Okay, the simple. This is the simple gene action, and the variation, okay, which will happen due to this particular gene action, the variation caused by this particular gene action will be your variance. ठीक है, additive variance or dominance variance. ठीक है, बताओ डाउट्स है क्या? Absence of dominance. Pooja, sir, you give that example, sir. Pooja, what you are asking? 10 मान के एक तो 15. The small a value will be 5, so there will be a 2a, so that it is 10. The capital A value will be 10, 10, so it is 20. ठीक है? How are you students? अरे कौन है भाई? In case of additive, the difference can be made as TT value remains fixed. Ready for 4 July. How it is fixable? अरे मैं how it is fixable मैं अभी तक नहीं बताया, ठीक है? How it is fixable, I will discuss now. How, why your additive gene action is fixable? We will discuss it. What not? Sir, not understand additive variance. The variation or variance which is caused due to additive gene action is additive variance. ठीक है? Additive gene action क्या है? Additive gene action is nothing but those are giving in cumulative effect. The both alleles are giving in cumulative effect. Okay, if it is capital A, they will give 20. If it is small a, then they will give 10, 5, 5, 10, 10, 10, 20. Okay, if it is capital A, small a, then they will give 15. If it is additive gene action. Okay, and if that particular gene action is present in the population and any variation is causing, any variation you are getting from this particular gene action, additive gene action, that variance we will call it as additive variance. ठीक है? वडनाड, दुर्गा प्रसाद, समझ गया ना अभी? Are you getting it? Additive genetic variance is fixable because it is represent the transmissible value of genes which can be accumulated by selection. ठीक है, मान सकता है. No sir, I had told you in the class. Please explain it by giving the example of yours. That is what I am saying. Yeah. Okay. Yeah, understand. Then सारे आ गया, सारे आपका gene action, 3 gene action. Then the variation which will develop due to this particular gene action, वो भी समझ गया, ठीक है? Then we will see, okay, before going into irritability, we will see why additive gene action is fixable. Okay, now the capital A value, we can say, whenever we are taking an example, we will try to see for single gene. Okay, but in the practical manner, in the practical thing, it's not about single gene. Okay, whenever you are talking about any trait, quantitative traits, it will be number of genes. But whenever we are dealing with example, we will take particular single loci. Your capital A will give 10, means it will contribute for 10 units. Your small a will contribute for 5 units. We can take like this. Okay, the capital A is your positive allele, small a is negative allele. Then, if it is additive gene action, if it is non-additive, means dominance. Non-additive is dominance. Okay, what you will do? Capital A into capital A, small a into small a. What about capital A? Capital A, if it is additive gene action, your value will be 10. Small a, value will be, 20. Small a, 10. Why? Your capital A value will be 10, 10. Small a, 5, 5. Okay. Then you will get an F1, capital A, small a. This is your F1. Okay. What about your F1 value? Can anyone? F1 value बताओ जल्दी लगा दो कमेंट में. 15. It is your 15. ठीक है. Then you will go for F2. You will go for F2. What about F2? Capital A, capital A, small a, small a, small a. Okay. Then what about value? This particular F2 in 20, 15, 10. Whenever in pedigree breeding and when you will start to do the selection in the F2 itself, F2 onwards. Okay, in F2 onwards you will start for selection. You will go to field and you will see which is giving highest unit. Okay, in the field you will go and see you are getting 20 in this particular individuals. Obviously you will select this particular plants. Okay, you will select this particular plant, then you will go for next generation. Okay, when you select this particular AA and you will go for next generation, then again it will segregate in the same way. So again you will get 20. Okay, once you select the population, once you select the particular individuals in that particular value, it is fixable. Okay, whenever you will next generation, again it will give 20. Clear? But in case of dominance, you can see 20, 10. Okay, capital A, small a. What about capital A, small a here? In case of dominance, whatever the value, maybe it is 15. Okay, maybe it is 16, 17. Okay, for example, you can take, okay, if it is complete dominance, it will give 20. Okay, we will take it as a complete dominance, 20. Then whatever, maybe there will be segregation now. So capital A, capital A, small a, small a. What about the value? 20, 20, 10. अरे ये इधर आएगा आपका प्रॉब्लम. ठीक है, आप अभी ये F2 है, अभी फील्ड में जाएंगे, देखेंगे सबको, सब प्लांट देखेंगे. अभी आप सब सारे इंडिविजुअल्स एवेल्यूएट करेंगे उधर जाके. ठीक है, you will go and you will see it is also giving 20, that particular individual, it is also giving 20. What you will do, based on the phenotype, you will select both these two. You don't know the genotype, right? Based on the phenotype you will select. Select तो किया है अभी. Okay, you will go, you will grow for next generation. Okay, your AA will give capital A. Your capital A, small a will segregate again. It will segregate like capital A, capital A, small a, small a, small a. Your value, it will be give 20. This will be your 20 again. This is also 20 and this is 10. Okay, if you see the average, 20, 20, 20, 60, 70, 70 by 4. What about 70 by 4? लगा दो, कोई कमेंट में लगा दो, पट लगा दो. 70 by 4. हाँ जल्दी. अरे बस एक घंटे हुआ ऑलरेडी, बस हम 20 स्लाइड्स खत्म किया है, अभी भी ज्यादा स्लाइड्स है. मेन प्लांट ब्रीडिंग मतलब मेथड्स में घुसना है अभी. हाँ रिमी 17.5. ठीक है, इधर कितना था? 20. अगेन इफ यू गोस फॉर नेक्स्ट जनरेशन, अगेन इट विल बी 20. अरे इधर तो ऐसे. नेक्स्ट व्हाट यू विल डू इन दिस पर्टिकुलर जनरेशन, you will select these 3. Again it will reduce, it will, yeah, generation by generation It will reduce. You are not able to fix the thing. Okay, that's what the basic. Okay, why your additive gene action is fixable. ठीक है, पता है ना, समझ गया ना अभी? बताओ बताओ, why additive gene action is fixable. सही हुआ ना अभी कनेक्शन? आवाज आ रहा है ना सही से? कोई कंफर्म कर दो अभी. Specific quality is nothing but it is due to segregation. We can tell segregation and recombination. Okay. I was also saying the same thing that AA will be fixed. Yeah. Okay. Pooja. Yes, sir. Homozygosity promote additive variance. Yes. Yes, sir. Okay. Yeah. This is what basic is. So what about irritability? This is again very important. Okay. Irritability is nothing but you are measuring the proportion of variation which is due to genotype. Okay. In irritability, you are not measuring the how much trait is transferring. Okay. If it is giving 100 kg, then in the next generation, from that 100 kg, how much kg it is transferring to the progenies? It's not like that again. Okay. The variation, okay, the variation present in the parent population, parents, okay, out of that variation, out of total variation, how much variation is transferring to the next generation? Sorry, that is your irritability. As I told, why we will talk about variation again? Variation is the basis for your selection. Okay, we need variation, right? So that's why we will always study about variation. You keep in your mind like this. Okay, the proportion of total variation in the character that is due to genetic cause. The proportion total variation, the proportion of total variation in a character, that is due to genetic variation is your irritability. Okay, you can see irritability is nothing but variation due to genotype to the total variation present in the population. Okay, this is your irritability. What about narrow sense? It is broad sense. Okay, this is broad sense. What about narrow sense? Narrow sense irritability includes only additive genetic variance. Okay, your variance, genetic variance is again subdivided into additive, dominance, and epistatis. Okay, but we don't need the dominance and epistatis in the selection. For selection, only your additive genetic variance will respond. Okay, as I told, okay, in the previous slide, why it is fixable. Okay, wherever you are talking about selection, okay, you will always talk about additive genetic variance. Where you will talk about dominance variance then? Batao? I am telling that wherever we are talking about selection, we will talk about additive genetic variance. Why? Your additive genetic variance will respond to selection, so that we can fix by selection. That means whenever you are doing selection, okay, if you want to get an accurate value, or if your selection is very much accurate, then you should target additive genetic variance. But when you will talk about dominance variance, then there is no need of dominance variance. Then what is the importance of dominance variance? छोड़ सकते हैं क्या उसको ऐसे? हाँ, अरे भाई लगा दो, बताओ कुछ। गुड गुड, heterosis. Okay, if it is dominance variance, then we can exploit heterosis or hybrid breeding. That's why I'm dealing these before starting breeding methods. Okay, we should know what are the variance, what is heritability. Okay, your narrow sensitivity is nothing but your VA out of VP. Okay, but your broad sense is VG by VP. If you want to tell which breeding method you should follow, then by seeing broad sense you can't easily tell. Okay, you should calculate the narrow sense irritability. Okay, if narrow sense irritability is very much high, then you can easily tell that it is controlled by additive gene action. The variance is due to additive gene action, so that you can exploit the breeding methods which are through selection methods like pedigree selection, okay, bulk breeding, anything which are selection. But where there is an your narrow sense irritability is very much low, that means your VA is low. If VA is low, then that obviously tells that your VD is more, so that you can go for hybrid breeding, heterosis breeding. What about co-irritability? Co-irritability is nothing but it is joint transmission of a two character, okay, two traits. Genotypic covariance of X1, X2. X1, X2 are two traits. Okay, whenever you are talking about irritability, for example, for yield we are talking here. Okay, for example, in co-irritability, we are talking about two traits simultaneously. Okay, the variation in that particular two trait jointly, that is your co-irritability. Again, the formula is same, that genotypic covariance between X1 X2 to the phenotypic covariance between X1 and X2. Just what you should know is co-irritability is nothing but we are talking about two traits at a time. Okay, joint effect of the two traits. Okay, what is the difference between broad sense and narrow sense? Okay, what proportion of observed variation is due to any genetic differences? What proportion of the observed variation is due to heritable genetic differences that respond predictively to the selection? That means it is which is responding to the selection, that is your relative genetic variance. Okay, that is your narrow sense irritability. The variance components, additive, dominance and epistatis. Okay, it will include your VG will include all these three. But in case of ER, your VA will only tell us about additive genetic variance. What it represents? The total genetic contribution to the phenotypic variance. It reflects the overall genetic control of trait. It reflects portion of genetic variation that consistently translate from the parents to offspring. In case of broad sense also, it will inherit. Okay, your VD, your VI, epistatic, dominance, they will also inherit to the next generation. But the thing is consistently. Okay, consistently your additive variance will transmit. Okay, predict response to selection. It is poor predictor. It is excellent predictor for genetic gain. Okay, as I told, VD and VA are generally not fixable. Your VA are fixable. Where we will use broad sense irritability, where there is no segregation. Okay, if it is a segregation, then you should also calculate the VD, VI. But when there is no segregation, it is your population or your material is very purely homozygous condition, then there is no question about your dominance, epistatis. You know, it is completely due to additive variance. So you can easily directly calculate your VG. There is no need to differentiate or distinguish between dominance, additive and everything in homozygous material. Okay, initial germplasm evaluation. Okay, where you want to know is there any heritable trait or not. Then you will go into deep in the next studies. In the initial studies, you just need broad sense heritability. And evolution of homozygous parents or inbreeds, where there is no other components. Okay, you just need some variance due to genetic factors. There you can use broad sense heritability. In case of animal breeding, where you are not able to cross and you want to see segregating material. In animal breeding, it is very difficult. There also you will go for broad sense heritability. But where you will go for narrow sense heritability, sexually breeding programs, okay, like self-planted, cross-planted crop plants in segregating population, majorly in segregation population. Okay, in non-segregating population homozygous material, it will go for broad sense heritability. In case of segregation material, where there is an chance of your dominance variance, epistatic variance, okay, that is in segregation material, there you can go for narrow sense heritability. A difference. So now, based on heritability, okay, if it is highly heritable traits are nothing but your qualitative traits, okay, your disease resistance, those are highly heritable. There is no environmental effect, right? समझ रहे हैं ना? If it is disease resistance plant, it will transfer that particular trait to its progeny. It is completely heritable, highly heritable. There is no environmental effect. In that case, okay, the direct selection, you can go for direct selection and based on individual plant performance. At that point of time, you can go for mass selection. For phenotypical, you can easily select the plants. Then pedigree method. Okay, why? In pedigree method, from F2 onwards, you will start for selection. If you are choosing any plant in the F2 itself, that means you are very confident that your F2 plants is expressing that trait in highly heritable manner. That's why we will go for selection from F2 itself. Okay? So you can go for pedigree in case of highly heritable traits, in case of seed color, in case of any oligogenic trait, for example, disease resistance, you can start to select from F2 itself. It is controlled by single genes again. Then backcross method. Okay, if it is highly heritable, then you will go for backcross. You can just transfer the trait and marker assisted selection, foreground selection by using markers which is linked to your trait. Then you will also go for marker assisted selection if it is highly heritable. Then what about low heritable traits? Yield. Whenever I am talking about low heritable trait, you should go for yield. You should think yield in your mind. Okay? Class is fully English mode. अरे शिनचैन, आपको क्या चाहिए बताओ शिनचैन? अरे इंग्लिश और हिंदी मतलब क्या डिफरेंस होगा? कॉन्सेप्ट तो समझना, मतलब आपको पता होना चाहिए हाईली हेरिटेबल ट्रेड्स में कौन सा ब्रीडिंग मेथड यूज करना है, फॉलो करना है, लो हेरिटेबल ट्रेड्स में कौन सा फॉलो करना है, बस वो पता चलना है ना. बस आपको कंसंट्रेट उधर करो, कंसंट्रेशन उधर रख दो, ठीक है? सिंचन, प्रॉब्लम हो रहा है क्या? सर यू कैन कंटिन्यू विथ इंग्लिश. इंग्लिश मोड. सिंचन, आप तो आपको सब चाहिए. मैं एक क्वेश्चन पूछा था आपको, हमारा कोर्स में एनरोल हुआ है नहीं हुआ है, कुछ मार्क टेस्ट में है नहीं हुआ है, वो क्वेश्चन पूछा था, आप नहीं बताया था आंसर. मैं क्यों पूछ रहा है, आपको एक हमारा कोर्स दे देंगे इंग्लिश, हिंदी इंग्लिश मोड, इसलिए पूछ रहा है. आप इतना दिन से मेरा लाइव क्लास अटेंड कर रहा है, इसलिए कुछ तो आपको देना है ना. मार्क टेस्ट में हूँ. मार्क टेस्ट में तो कुछ नहीं दे देंगे आपको सब ऑलरेडी पता है. लो हेरिटेबिलिटी ट्रेट्स, इसमें क्या करेंगे लो हेरिटेबिलिटी ट्रेट्स में? ओके, व्हेन देयर आर ट्रेट्स आर लो हेरिटेबल, ओके, आप डायरेक्टली उस ट्रेट को सिलेक्ट नहीं कर सकता है. फॉर एग्जांपल लीड, ठीक है, क्या कर सकते हैं, उस इनडायरेक्ट सिलेक्शन करते हैं. कैसे इनडायरेक्ट सिलेक्शन कर सकते हैं? हाउ वी कैन गो फॉर इनडायरेक्ट सिलेक्शन? आपको पता है यील्ड है ना, उसके आसपास कुछ ट्रेट्स है. फॉर एग्जांपल द स्पाइक, इन केस ऑफ वीट आई विल टेल द स्पाइक लेंथ. इफ इट इज मोर स्पाइक लेंथ, obviously you can expect more yield. If it is the number of spikelets in the wheat, if it is more number of spikelets, you will get more number of grains that will ultimately increase your yield. These are indirect traits. Okay, so you can go for the selection for these traits. We'll call it as a indirect selection. Okay, indirect selection of correlated traits. These are correlated. If your number of grains are increasing, that means it will obviously increase the yield. Okay, in low irritable traits, we can go the selection like this. But which are the methods you can follow? Family selection and progeny testing. Okay, in case of low irritable traits, as I told, you should always go for progeny test. Okay, by seeing the phenotype itself, you can't tells that it is due to, again, it is due to genotype or phenotype. If you want to know it is वो genotype से आ रहा है और phenotype से आ रहा है वो trait, इसको मतलब पता चलने के लिए आप progeny test करना है ये, ठीक है? Progeny test, it is one of the breeding method. Progeny test is nothing but your year to row method. Your recurrent selection also progeny testing. Okay, we'll see what exactly those methods are. Then your recurrent selection, as I told, bulk breeding. क्या करते हैं बल्क ब्रीडिंग में? शुरू में कुछ नहीं करते हैं हम, कोई बस प्लांट सेलेक्ट करके बल्क करके जाता रहता है F6 F7 तक, ठीक है? उसके बाद मटेरियल होमोजाइगस हो जाएगा. When your material got homozygous after some F7 F8 generation, then you will start selection. Okay, due to low irritability. When your trait is low irritable, you are directly not, you can't directly select plants from F2 itself. Okay, you should go up to till some homozygosity appear. Then you can go for selection. That we will do in bulk breeding in your SSD and genomic selection. Genomic selection is one of the recent breeding method. Okay, for low irritability traits. We will calculate each and every marker effect. Okay, that means again it is completely using markers. We will calculate the effect of markers. Based on that, you will select. If it is yield, then you will see which are the markers which are responsible for that particular yield. Okay, based on that, you will select. At this point of time, you should know for high irritable traits which are the breeding methods, for low irritable traits which are the breeding methods. कोई पूछ रहा था ग्रुप में क्विकेस्ट मेथड ऑफ प्लांट ब्रीडिंग, ठीक है? उसमें ऑप्शन था इंट्रोडक्शन, म्यूटेशन, ठीक है? एक चीज मैं बता सकता हूँ अभी इधर, ठीक है? ये क्विकेस्ट मेथड ऑफ प्लांट ब्रीडिंग, फास्टेस्ट मेथड ऑफ प्लांट ब्रीडिंग, ये तो क्वेश्चन है ना ये तो कौन फ्रेम करता है ना, मुझे पता नहीं वो कौन है, ठीक है? वो क्वेश्चन ऐसे नहीं चलेगा अभी. If it is quickest method, then there will be so many. You can also go for introduction, you can also go for selection, you can also go for mutation, ठीक है? वो तो ऐसे नहीं आएगा क्वेश्चंस, ठीक है? वर्डनर ऑलरेडी लगा रहा है इंट्रोडक्शन, ठीक है? उतना सिंपल तो क्वेश्चन ही नहीं है मेरा हिसाब से, ठीक है? If they ask, okay, introduction is not an actually the any plant breeding method. It is just one innovation, okay, where we can introduce directly the plants. And again, the mutation is not also not a quickest method. Okay, you have to treat the radiations to your plants, then M0, M1, M2, then you have to evaluate the plants based on your target trait. Then you can go for multi-location trial. अरे भाई कुछ ऐसे कोई मतलब क्विकेस्ट मेथड नहीं है, ठीक है? अभी बता सकते हैं मार्कर सेट सिलेक्शन, दैट इज आल्सो क्विकेस्ट मेथड. द क्वेश्चन इज नॉट दैट मच relatable. Okay, don't worry, they will not ask you these kinds of question. Okay, those are like before 2015, 2013, 2014 questions. Okay, when there are one liner questions. Fastest and quickest both are same or not? अरे छोड़ दो यार, वो सारे कॉन्सेप्ट में लगा दो आपका दिमाग. सारे प्रीवियस ईयर का 6-7 साल का प्रीवियस ईयर क्वेश्चन पेपर देख रहे हैं ना? हाँ, किसी में पूछा है क्या फास्टेस्ट कौन सा है, क्विकेस्ट कौन सा है, हाँ, मॉडर्न कौन सा है, ट्रेडिशनल कौन सा है? अरे छोड़ दो यार सारे. Don't follow different books for questions. And also I will try to remove those questions from our test series. Okay, मैं तो सही से नहीं देखा है वो सारे टेस्ट सीरीज, वो सारे निकालना है पहले क्वेश्चंस, आपको वन लाइनर क्वेश्चन, ठीक है। दैट्स इनफ, राइट, बट इट्स गुड टू नो सर। अरे भाई मैं बता रहा हूँ ना, this is unrelatable question. Okay, we can't tell that if they give both mutation and introduction, then it is again confusion, right? You just leave those things, they will definitely not ask. ठीक है, अभी इधर आ गया, ये तो कॉन्सेप्ट है, ठीक है, इसको आप सही से अंदर घुसना है, ठीक है, आपको दिमाग में अंदर डालना है ये सारे। अभी बताओ, देयर विल बी 3 वर्ड्स, इसमें कंफ्यूजन है, ठीक है, जेनेटिक एडवांस, रेस्पॉन्स टू सिलेक्शन, जेनेटिक गेन, क्या है ये सारे तीनों, ठीक है, बताओ अभी। अंशुल, नूतन, बताओ। इट्स ओके, इट्स ओके दुर्गा प्रसाद, आई एम नॉट मेकिंग यू फील बैड, जस्ट आई एम टेलिंग, ज्यादा उसमें टाइम नहीं मत लगाना, फास्टेस्ट और क्विकेस्ट मतलब लगा, मतलब जानने के लिए क्यों उतना टाइम वेस्ट कर रहा है, इधर लगा दो, जेनेटिक एडवांस क्या है, जेनेटिक गेन क्या है, इसमें लगा दो, सिलेक्शन डिफरेंशियल क्या है, ये तो पूछ सकता है क्वेश्चन, ठीक है। आई एम आस्किंग सिंचन, जेनेटिक एडवांस, जेनेटिक गेन, रेस्पॉन्स टू सिलेक्शन। नेक्स्ट ईयर तो प्लांट ब्रीडिंग बुक भी आएगा, उसके बाद ना आपको कोई डाउट नहीं रहेगा, उधर तक थोड़ा डाउट्स तो रहेगा, कोई नहीं, मतलब किसी को एक्सप्लेन करने के लिए पसंद नहीं है क्या ये चीज तीनों, कॉन्सेप्ट तो सिंपल है, ठीक है। these 3 words, genetic advance, response to selection, genetic gain, ठीक है, at the end of the day these 3 will give same meaning, but how we are expressing it, ठीक है, how we are putting in different words, that will be matter here, okay. All will be improvement, okay, all will be improvement in the selected progenies, but how we are expressing it, that will matter. Kavita is telling advance mean improvement in genotype value of selected plants over population. The difference between the mean value of parents and the mean value of progeny of called genetic gain. Genetic advance speaks to improvement. ठीक है, ठीक है, छोड़ दो, इधर आ जाओ, I will tell you. This is your parent population. Okay, this is your parent population. Population, when you are talking about population, it contains the numerous values. So that's why you will get a value, the graph like this. Okay, all the plants are giving the same value, then your graph will be like this. But it's not like that, na? It is population, normal distribution again. The traits will follow continuous variation. So that's why your graph will coming like this. Your mean value, the average performance of the population is the mean value. Okay, the original population. Then you will select the extreme plants. You will always select extreme plants, right? Okay, when you are talking about any trait, you will always go for highest value individuals. You will select some 5% of the population, one of the extreme, which are giving high yield, for example. Okay, this is the, we will call it as a selection intensity, at how much stringency you are doing the selection. Okay, you are doing 1% of the population, you are selecting only 1% of the population, then it is high stringent. If you are selecting 10% of the population, little bit low stringency. Okay, when you are having good skill and when you are able to target that particular individuals, then you can go for high stringency, that means less than 5% of the population. Okay, you will select some plants, this is the mean value of the base population. Then after selection, you will again you will plot a graph, then you will graph will comes like this. This is the again average performance of the selected plants. Okay, whatever the improvement you got, it's not an improvement, the difference between the mean value of original population and the mean value of selected population. That is your selection differential. Okay, mean value of selected population minus mean value of original population. This is your selection differential. Okay, yes. Then ये तो हो गया selected differential. Then you will grow this particular selected individuals in the next generation. Then what will happen? Then again there will be one curve. This is the next generation curve. Again there will be mean value. Okay, anything. Okay, anything that you can expect now from the mean value of original population and the mean value of progeny of selected individuals. This is the progeny of the selected individuals. Okay, what we will call? This is your response to selection. How much it is responding to your selection. Okay, this is nothing but your response to selection. That is nothing but mean value of progeny, the selected progenies to the original population. Okay, this is your response to selection. The actual improvement you got. The actual improvement you got through the selection. Okay. Next, what about genetic gain? What about genetic gain? Genetic gain बाद में देखेंगे. Response to selection हो गया, ठीक है? The actual improvement through the selection. Next, what about genetic advance? In case of genetic gain, there will be two words: expected genetic gain, realized genetic gain. Realized genetic gain is nothing but your response to selection. Okay, how much you got through the selection. What is expected genetic gain? That means before doing the selection, by using some parameters, you can expect the gain, the same gain again after the selection. How much you will get? Okay, you can expect before doing selection. That is your genetic advance. Okay, genetic advance is nothing but expected improvement from the selection in one generation. How much you can expect by using some parameters. Your parameters are nothing but, okay, genetic advance. Your parameter is nothing but K, sigma P and heritability. Okay, KPH. You know K is nothing but your selection intensity. I value or K, both are interchangeable, both are same. The stringency. Okay, then the value I or K, it is standardized value. Okay, the number of portion you are selecting, the number of portion of plants you are selecting is indirectly proportional to the K value or I value, the selection intensity value. Okay, you are selecting more number of individuals. That means the stringency is very low. If you are out of 1000 plants, you are selecting 100 plants. अरे भाई किसी को भेजो फील्ड में, आराम से वो 1000 प्लांट्स में आराम से 100 प्लांट्स उठा सकते हैं ना, अच्छे से देख के. सिलेक्शन इंटेंसिटी उतना नहीं है, स्ट्रिंजेंसी उतना नहीं है, उसको पता नहीं है उतना अच्छा से. आप जाओ एक ब्रीडर, ठीक है, आउट ऑफ 1000 प्लांट्स यू विल ट्राई टू सेलेक्ट ओनली 10 प्लांट्स. You don't want to waste these resources for the next generation. You will clearly see and you will only select some 10 plants. Okay, the number of plants are less. That means your selection intensity is high. The standardized value, that I will be increasing. Okay, that's what your proportion of selected plants is indirectly proportional to the selection intensity. Okay, IRK. When there is less number of plants are selected, your selection intensity will be high. If it is your selection intensity is high, that means the number of plants are selected very low. your selection intensity will high. If it is your selection intensity is high, then obviously you are getting the good genetic advance. Okay, this is the value of k. Value of k. Then your phenotypic variance, sorry, standard deviation. Okay, the how much variation you are having in the population, and the heritability of the trait. Okay, there should be a good heritability. This is your genetic advance. By this formula, you are expecting how much improvement can be done. Okay, response to selection is nothing but how much the real value, how much it is responding for your selection, the practical value. Your advance is expected value, and your genetic gain. Genetic gain is nothing but, okay, the same value, your heritability and your variation due to genotype and intensity. But over time, you will calculate the gain per year. Per year, how much the gain you are getting. Okay, the rate of change in the genetic gain. When you are including time, that is your genetic gain. Okay, and also we will call this particular response to selection, that is your RS is the mean of progenies of selected individuals and the base population. We will also explain in the term of H square S. H square S is nothing but you are replacing this particular intensity and phenotypic variance, such that standard deviation, with the selection differential. Since your selection differential is selection intensity into standard phenotypic standard deviation. Okay, if you, for example, if you want to replace this particular intensity and standard deviation, you can replace with the S, so that your response to selection will S square S. That is your, we will also call it as a Breeder's equation, which is given by Lush. Okay, Breeder's equation. Clear? समझ गया ना? एक simple table में मैं try करता हूँ अभी. First one is response to selection. As I told, the actual change in the population mean after selection, the actual change, that is nothing but your R is equal to H square S. Again, it is nothing but the mean value of progenies of selected plants minus mean value of original population. And what about S? Your S is nothing but mean value of selected individuals between the original population. The first one is your selection differential, and the next one in the next generation, that is your selection response to selection. ठीक है, वही है response to selection. Measures realized improvement. Then genetic advance, the expected improvement. Okay, you are expecting it before selection using some parameters. Expected improvement from the selection in one generation, that is your KPH. Okay, selection intensity into phenotypic, sorry, standard phenotypic deviation, and your heritability. It will predict the selection process. Okay, that means, yeah, we can do selection by before calculating the values. Then genetic gain, improvement achieved per unit time or generation, per unit time, per year. You are including one extra term, that is time year. Okay, nowadays this is very important. Okay, wherever we are talking about genetic gain, okay, we are introducing T, the time. It is important, right? Yeah, anyway you are getting improvement, but in 10 years, then what's the need? Okay, for every year you should get that particular improvement, right? Measures the breeding progress over time. This is one thing you should remember in this particular formula. Okay, we will include R. R is nothing but accuracy. Okay, yeah, we will replace this particular heritability with R value. That is accuracy. That means that is nothing but correlation. Correlation between your additive variance and the phenotypic variance. How that is correlated, your additive variance, and yeah, it is your additive variance. R is accuracy. Okay, I think, I hope now it's clear. Sir, for good genetic advance, we go for high irritability or narrow. High irritability. Broad sense or narrow sense? Array, whenever you are talking about genetic advance, you should always talk about narrow sense irritability, not broad sense. Okay. Sir, can we say that genetic advance occur in parent and genetic gain occur in progeny? अरे भाई दोनों progeny मतलब genetic, whether it is genetic advance, whether it is genetic gain, okay, when we will compare that in the progenies with the base population. Okay, as I told again, all three are having same meaning, but there will be some different expressions. Okay, how we are expecting it. Response to selection, as I told, it is expected change. Sorry, actual change in the population. Okay, we are doing the selection there, the practically. So we are getting some improvement. We are calculating it. But in genetic advance, again it is improvement only, but we are expecting it before the selection by using some parameters, using formula. Genetic gain, again the same thing over the time. इतना है simple. इस फार्मूला इंपॉर्टेंट सर, इतना आप लोगों के लिए उतना नहीं पूछेगा. PhD में भी in PhD they will ask, but there is no need to confusion. That's why I gave 3 words. Okay, then there is no need to go and see books here and there. In BDC they not given clearly, and you will think that अरे इसमें भी सही नहीं है, इस पे भी क्लियर नहीं है. ज्यादा सोचने के लिए जरूरत नहीं है. ठीक है, अभी क्रिस्टल क्लियर. गुड. सारे बताओ, अभी सारे बताओ, सब बताओ. क्लियर है ना? इतना मतलब बेसिक, इतना क्लियर कोई नहीं बता सकते हैं. बताओ. ओके, नेक्स्ट बायोमेट्रिकल टेक्निक्स. ठीक है, अगेन आई एम टेलिंग, in case of biometrical genetics, biometrical techniques, you guys don't need to worry too much about it. In case of your SRF exam, then again there will be a minimum 10-20 questions will come from this particular part. For you guys, they will not ask about these things. If they are asking, maybe they can ask the scientist who given that. Okay, but they will not too much about it. Now we have saw that the variances, irritability, and the different thing, but how we can calculate those things? First, as I told, the simple due to the calculation of variance due to genotype through the RCBD, through that particular field experiment. But how we will estimate additive variance, dominance variance, उसके लिए ना planned crosses चाहिए. ठीक है, there is a need of planned crosses, so that you can estimate those variances, combining ability. ठीक है, उसके लिए ये biometrical techniques. ठीक है, the branch of genetics deals with statistical concepts. Again, procedure for study genetic principles. That is your biometrical genetics. But techniques, what are the techniques we are going to use in that particular branch? There is always 4 important divisions in case of biometrical techniques where you will use for plant breeding. One is assessment of polygenic variation. Assessment of polygenic variation or the diversity. Okay, among the population, how much variation it is having. To get some assessment or to visualize that one, you will go for metroglyph analysis, D-square statistics. Metroglyph analysis is like a graphical method. Okay, by using graphs, you can say that the variation, you can express that variation in the graphs. Okay, how much variation is present. And also D-square statistics, by using the calculated distance value between 2 different individuals. Okay, you can estimate the diversity. Okay, simple thing you should know. And who given it, the metroglyph analysis, who given? Anderson, right? The D-square statistics, महाबलिनिस. ठीक है, ऐसे मतलब साइंटिस्ट पूछ सकते हैं. मतलब कौन सा चीज के लिए हम यूज करते हैं? फॉर पॉलीजेनिक वेरिएशन टू असेस और टू असेस डाइवर्सिटी. इतना आपको बेसिक आइडिया होना चाहिए. डिटर्मिनेशन ऑफ यील्ड कंपोनेंट्स. Yield components are nothing but, as I told, your yields are having some correlated traits like plant height, spike length, the number of grains, the number of spikelets. ठीक है, कौन सा trait इनडायरेक्टली एसोसिएटेड है, कोरिलेशन, उसको मतलब समझने के लिए हम ये सारे यूज करते हैं, कोरिलेशन एनालिसिस, पाथ एनालिसिस, डिस्क्रिमिनेंट फंक्शन एनालिसिस. Okay, these are the techniques, okay, which it will give some idea for the selection of indirect traits. आपको उतना पता होना चाहिए. Then study of gene action, this is very important. Gene action are nothing but your additive dominance. ठीक है, मैं तो सिंपल बताया था, अनोवा से हम बस जेनेटिक वेरिएंस पता सकते हैं, मतलब पता चला सकता है. ठीक है, और थोड़ा आगे, ओके, इफ यू वांट टू आइडेंटिफाई एडिटिव जीन एक्शन, डोमिनेंस जीन एक्शन, उसके लिए मैं बताया था ना, प्लानड क्रॉसेस चाहिए. ये वही है सारे, डायलील क्रॉस, partial allele cross. Diallele cross are nothing but if you are having some 20 lines, you will cross among the individuals. Okay, how much is like in diallele cross? Okay, how much the possible crosses are possible in case of diallele direct crosses? For example, you are having some 10 lines. Okay, tell me how much possible crosses are possible, diallele crosses. Diallele cross by Yeaman 1954, GCA, SCA, D and H components. सारे छोड़ दो सिंचन, नहीं पूछेगा आप लोगों के लिए वो तो. ठीक है, आपको बस पता होना, मतलब पता होना चाहिए किसके लिए यूज करता है. ठीक है, वो सारे एनालिसिस, योर डायलील क्रॉस, पार्शियल डायलील क्रॉस, ओके, लाइन इनटू टेस्टर, quadriallele, biparental cross, these are like planned crosses. Okay, in diallele crosses you will go for all possible crosses. In case of partial diallele you will go for only selected classes. Okay, in line into tester there will be some lines, you will consider some are the lines and you will consider some are testers, male into female crosses. Okay, then in case of quadriallele you will go for double crosses. Okay, in case of generation mean analysis, you will analyze F1, P1, P2, F1, F2, okay, F3, back crosses. These are the different crosses, different generation. Okay, you will grow in the fields and you will estimate all these planned crosses. By using that you can go for study of gene action. ठीक है, कुछ वीडियो में कुछ गड़बड़ हो रहा है क्या? कुछ ವೈಫೈ ಹೋಗಕ್ಕೆ ಬರ್ತೈತೆ ಅಕ್ಕ, ಇದನ್ನ ಕನೆಕ್ಟ್ ಮಾಡಿ ಬಂದ ಅದ್ರಾಗ ಎತ್ತಿ ಈಗ. ಓಕೆ ಬಾಯ್. हेलो, आवाज आ रहा है क्या? बताओ. ठीक है, सही है क्या? अभी वॉइस सही है ना? वीडियो भी सही है ना? कुछ ब्लरिंग तो नहीं हो रहा है ना? ठीक है. Then assessment of varietal adaptation. Okay, how your variety, once you develop the variety, okay, whether it is stable, before you are releasing any varieties for particular agroclimatic zones, you will test for stability. Okay, whether it is suitable for all agroclimatic zones, whether it is stable in all around their environments, what is the stability of that particular variety. Okay, for to know those stability, again there will be a different stability analysis model. Okay, Everett and Russell model, Parkinson and Jinks model, those are again statistical models. Okay, they will just tell us about that your genotype is stable, if it is stable in which, how many environment which is stable. Okay, or it is having some responsive to the environment, that response we will call it as a G into E interaction. to know those things, there will be some different statistical tools. Okay, we'll call it as a stability models. Okay, again, these are the things. This much is enough for you guys. Okay, as my opinion, they will not ask you the scientist names also. Okay, if they are asking, maybe they will cover most basic biometrical techniques and who given those techniques. Okay, there will be again 4 different things for assessment of polygenic variation or diversity analysis, for determination of yield components, or it will be in the help in the selection of indirect traits and study of gene action and to assess the varietal adaptation. Okay, these are the different, again, scientists. Okay, you can just take a screenshot or whatever, do whatever you want. Carl Pearson is the person. Okay, he is the main person where he contributed too much towards biomedical genetics. You should know his name. And Fisher. Okay, and these are the fellows. They given different planned classes. Okay, the line into tester, diallele class, these are nothing but planned classes. That's it. The biparental mating designs, the different designs, these are like planned classes, and the statistical measures. But through those classes, you can estimate the additive and dominance variances. Okay, these are the different scientists which they given different designs and different biometrical techniques. Okay, now guys, this is very important section. Now we are dealing. Okay, from now onwards we will see the breeding methods. We will just clearly see what are the breeding methods, where you should use, applications and merits. Simple. And if it is very important, then we will see the schematic representation. ठीक है, अभी थोड़ा आप कुछ ब्रेक चाहिए तो ले लो 2 मिनट. ठीक है, कुछ शेयर कर दो सबको. व्यूज तो कम आ रहा है. पहले दिन तो 100 आ रहा था. अभी तो इंटरेस्टिंग पक्का. ठीक है, फ्रॉम नाउ ऑनवर्ड्स देयर विल बी इंटरेस्टिंग थिंग टू डिस्कस and it is very important for your exam also. Okay, अरे वो बोलने के बाद कम हो रहा है. As I told, मुझे तो व्यूज चाहिए. इतना तो थोड़ा कॉन्फिडेंस और एनर्जी आएगा ना. मुझे तो एनर्जी ड्रिंक नहीं चाहिए. बस अच्छे से लाइव सेशन में सारा ज्वाइन होना चाहिए. इसलिए हम मतलब ये स्लाइड बनाने के लिए, रेडी होने के लिए 2 दिन 3 दिन लगा है. ठीक है, that's why I am telling that you should be till the end of the class so that you will get some information. हाँ, मुझे पता है सारे अपलोड होने के बाद 2x 3x में लगा के वो भी कर सकता है. अरे भाई, थोड़ा एंजॉय भी करो लाइव क्लास में. ठीक है, आप डाउट भी पूछ सकता है. इतना 49 एक्चुअली कितना है? Okay. Anyways, till now, any doubts? We discussed the quantitative, qualitative traits. Okay, and the different variants. Why that variance is important? Why that gene action is important? As I told, based on that only we can move forward in breeding. And the low irritability, high irritability traits. Okay. Okay. Enjoying, sir. You must have gone to eat, sir. Everyone is coming. नहीं, मैं तो सारे खत्म करके डिनर करेंगे. ऑलरेडी इंटरेस्टिंग है सर क्लास. वो तो मुझे पता नहीं. मैं सारे पता नहीं क्यों कम लोग है और बोरिंग हो रहा है, पता नहीं मुझे. विदिशा घोष बता रहा है, इंटरेस्टिंग है, ठीक है। Everyone is taking energy, yes sir, yes sir, enjoying sir, the previous class has 3000 के, अभी वो तो 4000 चला गया पूजा सर, बट नो लाइक्स और कमेंट्स। मैं बताया है ना अभी, इस वीडियो मतलब पता नहीं कमेंट्स भी आना है, इसमें भी वो एक और डिस्क्रिप्शन बॉक्स में डाला है ना हमारा लॉन्चिंग कोर्स का वीडियो, उसमें भी कमेंट्स आना है, ठीक है। मेरा नाम भी ले लो, मुझे पता चलेगा, मतलब मेरा स्टूडेंट्स भी कुछ कमेंट किया है, ठीक है। उसके बाद ही पार्ट 3 आएगा, ठीक है। पार्ट 3 में फिर से कुछ इंपॉर्टेंट चीज है और हमारा स्ट्रेस ब्रीडिंग का पूरा रिवीजन करेंगे, ठीक है। कहाँ भी नहीं मिलता है वो, यू विल नॉट गेट एनीवेयर दैट पार्ट, एंड सम स्पेशल ब्रीडिंग टेक्निक्स, द रीसेंट इंप्रूवमेंट्स, द रीसेंट breeding methods, recent achievements whichever happened, everything will be, and regarding some genetic engineering part, markers, सारे पार्ट 3 में आएगा। वो तो मतलब इम्पोर्टेंट है, फिर से चाहिए तो आप, मैं बताया ना क्या वो करना है, वो सिस्टम कॉर्न इज इम्पोर्टेंट और नॉट। अरे सिंचन आप अकेला मेरा साथ इंटरेक्ट कर रहा है, सारे ले रहा है इंफॉर्मेशन, हाँ। We'll see everything which are the breeding methods, okay? Okay, 50. From now onwards, it is very important, okay? Be till the end. We will discuss all the breeding methods. First, you know the mode of pollination and reproduction plays an important role in the plant breeding. If it is self or cross, asexually, based on that only we will target different breeding methods. Okay, if it is self-pollinate crop plants, as I told, it will love homozygosity. Okay, it should tend to be in the homozygosity. Okay, if there is a selfing, so there will be increase in homozygosity, okay, generation after generation, so that naturally it should love, okay, it should love to be in the homozygous condition. So whatever the breeding methods we are doing, okay, at the end of the day we should target for homozygous plants. But in case of cross-planted crop plants, as I told, they tend to be in the heterozygous condition. They love heterozygosity. So whatever the end value, the end product, it should be in heterozygous condition. That's why in case of cross-planted crop plants, you are highly targeted for hybrid varieties, synthetics, composites, and also about population improvement. Okay, in case of asexually propagated crop plants, there is no meaning for the any selection methods, okay, like your pedigree, back cross, okay, there is very difficulty in crossing. So in that particular crop plants, okay, what we can do, we can go for clonal selection, mutation, okay, polyploidy, we can explore those kinds of breeding methods there. Okay, we will see each and everything. As I told, different breeding population, homozygous, heterozygous. फिर से मैं ऑलरेडी PYQ सीरीज में भी बताया है, ठीक है, व्हाट एग्जैक्टली द होमोजाइगस, हेटेरोजाइगस at the individual level, at individual gene level, ठीक है, at particular individual level, if there are so many number of genes, okay, capital A, capital A, capital B, capital B, capital C, capital D, small d, okay, this is one genotype, for example. At gene level, this is homozygous. Then small a, capital small b, small c, capital D. Again, at individual level, homozygous. If you cross these two, you will get capital A, capital B, capital C, capital D, small d. Again, at individual level, it is homozygous. But when we will talk about population, then you will go for homogeneous and heterogeneous. Okay, homogeneous is nothing but all individuals in the populations are similar in the gene level. Okay, if all are like this particular individual, then it is homogeneous. If all individuals are in this particular level, then those are homogeneous. You don't see that the gene level, okay, If it is one individual and against the second individual also same like A, capital B, capital C, capital D, small d. Okay, if you compare these two, if you when you are taking in the system in these two individuals, then you will tell these two are homogeneous. Yes, in the individual level, these are heterozygous. Where you can see like this, all individuals are similar, but at individual level it is heterozygous. In which population, okay, you can see like this. You can see that heterozygous and homogeneous. The condition is heterozygous and homogeneous when you consider these two. These are heterozygous at individual level, but as a population, those are homogeneous, similar. Yeah, where you can find this? F1 hybrid. Yes, good. Okay, this is what exactly? Homogeneous and heterogeneous are nothing but there will be a different genetic constitution in the population. One plant will be like this, one plant will be like this, one plant will be like this. That is your heterogeneous. Okay, when you consider this is heterogeneous, but when you consider this and this, both are homozygous in nature. Okay, both are homozygous in nature, but heterogeneous. Where you can see this, homozygous but heterogeneous. Yes, good, Sinchan. Multilines are homozygous and heterogeneous. These are multiline condition. Okay. If all are like this, then pure lines may be homogeneous and homozygous. Okay, these are the different populations. Classification of plant breeding methods based on the application in the crop improvement, general breeding methods. Okay, your pedigree, okay, your pure line, pedigree, bulk breeding, your year to row, okay, back cross. These are general breeding methods. Okay, what are special breeding methods? Special breeding methods are your mutation, okay, polyploidy, your idiotype breeding. These are special breeding methods. What are population improvement approaches? Population improvement approaches is your recurrent selection. It is your population improvement. In case of population improvement, you are not actually concentrating on individuals. Okay, it is all about population. You are trying to accumulate favorable alleles in the population. Okay, there is no need to worry about single individual there. Then based on hybridization, methods involving hybridization, methods not involved in the hybridization. Okay, where there is an already existing variation, okay, then there is no need for hybridization. Your mass selection and your pure line selection, okay, these are not involving in the hybridization. Okay, your year to row selection, these are the things where you will not go for hybridization. There is already a variation present. ठीक है, already variation तो ये population में आप बस selection कर रहा है उधर. As I told, variation is the basic thing you need. Already variation वो तो क्यों hybridization करना है फिर से, ठीक है? उधर डायरेक्ट वो जाके मास सिलेक्शन कर सकते हैं, प्योर लाइन सिलेक्शन कर सकते हैं. Methods not involving, sorry, methods involving hybridization, sorry, these are methods not involving hybridization. ठीक है, but methods involving hybridizations are your pedigree, backcross, bulk, your recurrent selection. ठीक है, all are different breeding methods. Next, methods of breeding in different crop types. Again, it's the thing is very simple. In self-planted crop plants, okay, your pure mass selection, pure line selection, pedigree, backcross, bulk, will follow those things. In cross-pollinated, again, mass selection is the same thing in everywhere, and your year to row selection, population improvement methods, those are your recurrent selection. Your recurrent selections are also as a progeny selection, okay? But there is some difference, I will tell you. And again, your hybrids, synthetics, composites, everything will comes in cross-pollinated crop plants. In your asexual propagated crop plants, as I told, clonal selection and mutation breeding, these are very two important methods where you can exploit the breeding in case of asexually propagated crop plants. First one is mass selection. Okay. In these things, in these slides are breeding methods, what you should know, okay, the procedure and the application. These are the two important things. Okay, maybe they can ask you in the statement wise questions or assertion and reasoning types of question. Okay, where in which breeding method you can expect good transgressive segregants, in which breeding method you can exploit greenhouse and polyhouse condition very effectively, in which breeding method your skill is most important. Okay, these are the very important thing you should know. First one is mass selection. Mass selection is very important. One thing I should correct here, in one of the PYQ, okay, while I am solving, there is some mistake I did. Okay, the thing is, the variety which is developed through mass selection, okay, it is very easy to identify in nucleus seed production or breeder seed production, in seed production plots. Okay, there is a statement like this. Okay, but it is very difficult to identify. That in that PYQ, as I think I told that it is easy to identify in the seed production fields, but it is very difficult since your mass selection varieties are developed through the phenotypically, yeah, nucleus seed production maybe. In the varieties which are developed through mass selection are, it is due by phenotypically we did, maybe there will be a segregation and there will be a no uniformity in the field, so that it is very difficult to identify. Okay, if it is very uniformity in the field, you can easily tell that yeah, it is very uniform, this is one variety. But if it is admixture, then it is very difficult, okay, to identify that particular varieties. You can see here mass selection, how we can do this is a source population. There is a population of maize, for example. You will just go to the field and you will select out of some 100 plants, you will select 10 plants. Okay, what you will do, you will just select 10 plants, okay, and you will bulk the seeds and you will grow next generation. Okay, then again you will go for field, then you will select again best 10 plants, then you will select the seeds, you will bulk it and you will grow for next generation. Then after some time, okay, you will just go for yield trial, then you will release as a variety. Simple. The phenotypically superior plants you will go and you will select and bulk the seeds and you will grow for next generation. Okay, this is the simple breeding method. Select and bulk the seeds of desired plants, then plants replicated in bulk, then you will go for release best performer. Simple mass selection. And you can also go for another mass selection for it is cultivar development, any variety development. There is a mass selection for purification of a cultivar. Okay, what you will do there in case of mass selection for the purification of cultivar? There is a source pollination. The source population is nothing but your variety, already developed variety. You will go for so those particular population and you will select some desirable plants. Grow about 200 plants, okay, or heads in row. Then you will rogue some of types. Okay, whatever you are not interested, you will just rogue them. You are purifying the varieties, the second objective of your mass selection. Okay, so that the remaining ones you will pool and you will bulk and advance. Okay, this is the one of the application of your mass selection, that is purification. In purification, you will rogue out the off types to maintain purity of existing cultivars that have become the contaminated by any mutations or any admixture or mechanical mixture. Another one is improvement of local variety. Okay, to improve the local variety, you will just go for mass selection. Okay, this is the simple two applications of your mass selection. Too blurry. अरे भाई आज तो क्या हुआ? Too many glitches. No, ये तो आज थोड़ा इंटरनेट का प्रॉब्लम हो रहा है. ठीक है, क्लियर है ना अभी? Purification of cultivar. In first one is cultivar development. Okay, mass selection. You will just select the good plants, then you will bulk, then you will go for next generation. Okay, then after some years, after 2 to 3 years, whenever you want to, whenever you feel that it is you are what you are expecting, then you will go for yield trial and you will release. But in case of purification, you are already having some variety. Okay, you are having some variety, you just have to purify that. How you will purify? You will just grow that particular population, then you will select some 500 to 1000 desirable plants. Desirable plants are nothing but you know already their variety, the characteristics. Based on that, you will select the plants which are having some your desirable variety lines. Okay, then you will sow those in the next generation. Okay, from heads in the rows means year to row method, like that. Then you will go for inferior progeny. If you see any inferior plants, then you will just remove full particular row. Then the remaining rows will be bulk and you will go for bulk harvest in the next generation. Okay, so that you can purify by removing that inferior plants completely. Okay, purification and improvement of local varieties. These are the two applications of mass selection. Okay, please explain again, sir. Right, फिर से लकी पटेल, अभी तो क्लियर है ना? क्लियर है ना? बताया, मैं अभी दोनों बताया. ठीक है, बताओ बताओ कमेंट में. Okay. Next, your pure line selection. Before you are going to pure line selection, you should know pure line theory. Okay, the pure line selection is based on the pure line theory. Don't confuse that both pure line selection and pure line theory are same. Okay, pure line selection is method used to develop a new variety by isolating a progeny of a single superior homozygous plants. Your pure line is nothing but the progeny of a single superior homozygous plant of a self-planted crop plant. Okay, that is your pure line selection. But before that, you should know pure line theory. It is given by Danish botanist Johnson in 1903. What you will do? You will take the princess variety of Phaseolus vulgaris in the market from the commercial seed lot. Okay, first what you will do? The initial selection. There will be seed lot. You will separate the lines. Okay, individual seeds of different size are selected to raise individual seed progenies. You will separate into 19 lines. Okay, so initially what you got? From the particular original seed lot, there is a variation in the seed size. So he tried to select from 1 to 19 lines. So there is selection is effective. If we grow the large seed, okay, the seed weight, if you, for example, seed weight or seed size, then you will get larger seed. Like that, if you select this lower seed size, then you get lower seed size. The selection is effective in the beginning, initial separation. Okay, since the variation is present in the original seedlet is due to genetic factors. Then what you will do in the next phase, the 19 pure line, okay, what you will do, you will separate the 19 pure line. You will develop pure lines, 19 pure lines. Then what you will do, for example, in that particular 13th line, you will do different seed weight classes. Once after pure lines are developed, like 19 pure lines, what you will do, among that particular pure line, the 13th line, you will try to separate based on the seed weight. 200 seed weight separate seeds, 300, 400 and 500. You will make the separate groups among the 13th line. Then you will grow for next generation. Then you will see the progenies. The progeny seed weight. अरे क्या हुआ, सारे प्रोजेनिस का वेट ना मीन वेट सेम ही आ गया फिर से. अरे ये तो 450 था, वो ग्रुप कर दिया, ठीक है, 200, 300, 400, 500 जैसे, ठीक है. उसके बाद प्रोजेनिस ग्रो करने के बाद ना वो ग्रुप किया था ना, वो सारे ग्रुप्स का मीन वैल्यू था ना, वो सेम ही आ गया. सेम ही मतलब अराउंड 450. So that's why he told the selection within the pure line is ineffective. First one is, among seed lot is able to separate into 19 lines. What that means? The selection in the original seed lot is effective due to genetic factors. But after in 13th line he tried to do the selection, but there is no improvement. Okay, there is no improvement or there is no classification at all. So what he told, selection within the pure line is ineffective. Then in particular line 1, what he did, sorry, in this particular 13th line he told whatever the variation, okay, you can see the variation within the pure line. Sorry, in this particular second experiment, what he did in the particular 13th line, he tried in the different seed size, 200, 300, 400, 500. Okay, so the variation, it is variation 200 to 500. That particular variation present in this particular 13th line is due to environment, what he told. In the first line, he tried to do the selection, the mean weight is 640. He tried to do the selection of largest seed, smallest seed for continuously for 6 generation. But at the end of the day, okay, again, when you take the mean seed, okay, again it is nearly to the same in the beginning of that particular selection. Okay, like 690, 640, it is 690 and 680, like that. Then he told the selection within the pure line is ineffective. Okay, the testing selection within the line. Okay, there are 3 more important points. The conclusion of pure line theory are, a natural self-fertilized population is a mixture of several different homozygous genotypes, the original seed lot. Because this variation has a genetic component, selection is effective in separating these types. He separated 19 lines. Then a progeny derived from a single homozygous plant is pure line. Okay, all individuals are genetically identical. Variation within the pure line is 100% environmental. Okay, the variation what he saw in the within the pure line, okay, within that particular 13th line, okay, mean seed weight is 450, but he tried to group into different weights. Okay, again, obviously there will be variation, but that variation within the pure line is due to purely environmental. Okay. Consequently, the selection within a pure line is ineffective. Okay, as it does not change the genotype. So that the selection within the pure line, largest seed, smallest, largest, smallest, after continuous 6 generation, then there is no improvement. Okay, so that selection within the pure line is ineffective. Okay, this is the basis for pure line selection. Okay, this is our pure line theory. What you should know: the proportion of completely homozygous plants, okay, at any particular generation is 2 to the power of m minus 1 divided by 2 to the power of m. This is actually, this is formula for completely the proportion of homozygosity. The proportion of homozygosity. The number of genes will not influence the number of homozygosity, the percentage of homozygosity. Okay, and also your linkage. Okay, whether it is there are the two genes or more number of genes, and those are in linkage or not linkage, will not affect the percentage of homozygosity. Homozygosity is nothing but at that particular the gene level. Okay, they will not affect. But when you are talking about completely homozygous plants, for that your number of genes will affect the n. Where when n will come into the game, the homozygous plants, completely homozygous plants. But when you are trying to explain homozygosity, that is that individual particular gene level, then there is no need of talking about the number of genes. But when you are talking about the complete homozygous plants, then you should mention that particular the number of segregating gene pairs, and here your linkage will comes into the picture. Okay, when there is a linkage, your number of segregating gene pairs will decrease. So okay, when there is a linkage in your particular population, the proportion of completely homozygous plants, okay, will increase. The more the number of genes are in linkage, okay, the proportion of completely homozygous plants will be very faster. Okay, since your n, when you increase your n, it is directly proportional, right? When your number of genes are more, the number of genes segregating are more, the proportion of completely homozygous plants will get decreased. Means it is low. Matlab there is not that much fast. Okay, when there is a more number of n, the number of segregating genes. And what about m? M is your number of generation of self-pollination. When you are talking about the homozygosity, you can say 2 to the power of m minus 1. Okay, your m, for example, your m is 1. The number of generation of self-pollination is 1. If you take 1 as your m, then 2 to the power 1. 2 to the power 1 is 2 minus 1. 1 by 2. 1 by 2 is 50%. For every generation of selfing, there will be a 50% increase in the homozygosity. Okay, but when you are talking about homozygous plants, there you should consider the number of segregating gene pairs, and your linkage will affect. Okay, that you should know. Then why do pure lines degenerate over time? Why? One reason is maybe mutation, and maybe due to mechanical mixture. These may be the reasons for your degeneration of pure lines. ठीक है? प्योर लाइन सिलेक्शन डील होने के बाद थोड़ा मार्केटिंग करेंगे, ठीक है? What we will do in the pure line selection? There is only one difference between the mass selection and pure line selection. The bulk. You are going to bulk the seeds in case of mass selection. अरे इधर ऐसे नहीं करेंगे. फील्ड में जाएंगे, there are 1000 plants. Okay, in that 1000 plants you will select some 200 plants. सेलेक्ट होने के बाद, सेलेक्ट करने के बाद, ठीक है, 200 प्लांट्स के सेलेक्ट करने के बाद एक एक प्लांट का सीड्स है ना, नेक्स्ट जनरेशन में एक एक लाइन में सेपरेट ग्रो करेंगे. 200 सेलेक्ट प्लांट सेलेक्ट करने के बाद सीड्स तो बल्क नहीं करेंगे इधर. वी विल नॉट गोइंग टू बल्क द सीड्स, वी विल कीप इट एज ए सेपरेट seeds of individual plants. Then we will grow the individual rows of that particular plants, plant progeny rows of superior plants. Then we will compare. Then again we will reduce this 200 into some 25 to 50. Then we will reduce for 15. Then you will go for preliminary yield trial. Okay, we are maintaining the purity here. Okay, we are going to maintain the purity. We are not going to bulk the seeds. What are the application? Improvement of local varieties again. Okay, if there is any mixture, what we will do? We will just select the superior plants and we will grow individual plant progeny rows. Then we will keep it as separate seeds. We are not bulking it. Selection in introduced varieties. If you introduced any variety from outside, then you will do some selection, then you will release. Okay, improvement of old pure line varieties. For improvement of old pure line varieties also, you can use particular pure line selection. These are the particular application: mass selection and pure line selection. What's the difference? Mixture of pure lines, your mass selection, you are bulking it. That's why it is single pure line. Less uniform, shows genetic variation. And the another thing, okay, you can see here from 1000, 200, then 200 to 25 or 50, then 15, then 10. Okay, at this point of time again you are having 10 lines in advanced yield trial, 10 lines. Then you will choose any one of particular superior line and you will move forward. You don't merge this bulk these 10 lines. Anyway, you are end of the particular program, so you are having some 10 superior lines. You can bulk and you can base the trial ko. ऐसा नहीं. You will only select any one of the line. If you select two lines, if you are selecting two lines also, you are maintaining, you are pushing to trial as a separate two lines only. There is no bulking here. Okay, that's why it is having purity. You can see here, highly uniform. Progeny test generally not carried out, but here mandatory and careful. You are doing the progeny rows now, you are growing as a progeny rows. Wider adaptation, greater stability, but here narrow adaptation means it is completely homogeneous material, so narrower adaptability. Both you can use in both self and cross planted, but here use it in only self planted crop plants, but sometimes cross. That means when you are doing inbreed development, you will go for selfing the same as pure line selection. You will go for in case of inbreed development, you will do the same procedure. Okay, difficulty to identify in seed certification, the point. Okay, it's very difficult to identify in the seed production, seed certification, since there is no uniformity, but easily identified in the seed certification. Okay, typically for 5-7 years, ये तो 7-8 years टाइमलाइन तो नहीं पूछेगा. अभी तो ये ब्रीडिंग मेथड सारे ऐसे ट्रेडिशनली नहीं कर रहा है. ठीक है, अभी तो थोड़ा एडवांस ब्रीडिंग मेथड्स आ गए ना, ये तो पूरा ट्रेडिशनल हो गया अभी. तो प्रिंसिपल तो सेम ही है. Before going to pedigree selection, okay, as I told, now we are available, our mock test will be, the test series will be available for $399. Whoever not yet enrolled to our test series, you should go and purchase now itself. In that particular test series, there will be so many mock tests available already, and also you will get these marathon PDFs in that particular course, and there is already the video discussion of that particular each and every mock test. There will be a separate classes which we have already discussed. Okay, you will get so much benefit, and the course will be, master course will be 1,999. Anyway, I will not recommend this since there is a less time. You should buy this particular test series. Then ASRB and ICAR PhD course, this is for 365 days. We already covered each and everything as per the syllabus of ASRB, ARS. ठीक है, you can also enroll this particular genetics and plant breeding course. And for next year, this for 2028, 8,999 and 4,999 for next year, that is for 2027 course. Okay, anyway already मतलब वो enroll हुआ है ना, उनको already पता है, ठीक है, वो सारे benefit already ले रहा है, ठीक है, आप लोग भी feedback ले लो आपका सीनियर्स मतलब कोई किसी से, ठीक है, जल्दी जल्दी एनरोल करो. नेक्स्ट विल सी अबाउट पेडिग्री सिलेक्शन. कौन दिया था पेडिग्री सिलेक्शन? यस विजयाली. मास सिलेक्शन टू बल्क बट द प्योर लाइन आर नॉट बल्क. यस, पेडिग्री सिलेक्शन कौन दिया था बताओ? बताओ बताओ जल्दी. लव लव, अरे स्पेलिंग सही से बताओ कोई. रतन सिंह बुलार लव लव लगा रहा है. लव लव. ठीक है, the pure line selection mass selections when we will use when there is already existing variation. We are not doing any, we are not creating any variation. But when there is no variation in the population, original base population, you should create some variation, right? So what we will do in case of that particular scenario? We will first cross any two parents P1 into P2. Then you will get F1, the all are similar. Then you will go for F2. From F2 onwards, okay, you will use your skill as a breeder. You will use your skill and you will try to select some plants based on your experience, based on the visual appearance, phenotype. You will try to select the plants. Okay, around 2,000 to 5,000 plants which are in the F1. You will try to select some 100 or 200 plants. You will grow that particular F2 individual as a single row. Particular F2 individual as a single row, and you maintain the record also from which F2 plant the which row has came. You will maintain pedigree of that particular thing. Then again you will select some best superior rows and you will grow in the next generation like that. Okay, after from F6 generation you will go for, from single line you will grow 2 line or 3 line. Okay, progeny, family progeny rows. Then out of those you will see for each and everything the yield, quality, okay, the disease and pest, everything you will see. Then you will go for preliminary yield trial. Like that you will reduce the number of lines. At the end you will go for any good superior line, okay, as a cultivar release. The simple thing you should know here, okay, you will start selection from F2 itself and you will create variation by crossing, hybridization, and you will maintain the ancestry. Okay, if after the release of this particular line, you can easily go back and see from which F2 plant it all started. आपको पता रहता है, ठीक है, कहाँ से शुरू हुआ था ये, इसका बर्थ, ठीक है, वो F2 है ना, कौन सा F2 प्लांट से आया है, सब पता चलेगा, सब रिकॉर्ड आप मेंटेन करेंगे इसमें, ठीक है. What are the key features are ancestry records, and the base population is by creating the variation P1 into P2 and phenotypic selection from F2 onwards. You will use the skill, okay, for breeder to exploit his skill, which is, this is the best breeding मेथड, बस इतना पता होना चाहिए, इतना रखना है दिमाग में, ठीक है। पेडिग्री सिलेक्शन, व्हाट आर द एप्लीकेशन, सिलेक्शन फ्रॉम सेग्रीगेटिंग जनरेशन, not like in pure line, it is from the base population, the already existing variation. But in case of pedigree method, you are selection from the segregating population. After creating the F1 and F2, it is getting segregating from that population. You are doing the selection, correction of defects in existing varieties. Okay, if you want to correct any defects, okay, you can cross P1 into P2, then you can combine those into single variety. Okay, this is what combination breeding is. Your P1 is having good yield and P2 is having good adaptability. You want to merge them both into single plant, single individual. Your pedigree is the best method to do the combination breeding. Development of superior recombinants, that is your transgressive breeding. Superior recombinants are nothing but, okay, better than your parents. Your parents is giving, for example, your parent 1 is giving 20 unit of yield, parent 2 is giving 30 of unit of yield. You are segregating material, okay, in case of your F2, it will extreme of the both the parent, maybe less than 20 or more than 30, that is your transgressive segregants. Okay, due to the different gene combination and different recombination, okay, you will get the extreme of this particular two parents. Those are your transgressive segregants. Those you can exploit through the pedigree method. Then development of pure line varieties. Again, at the end of the day, what's your the product? The end product is your pure line. Okay, after the crossing and everything, the procedure will be same, similar to your pure line. Okay, you are maintaining the single line as a separate individuals. There is no mixing here. Same as your pure line. Okay, at the end of the day, the product will be pure line variety. Okay. Then when irritability is high, as I told, you will start your selection from F2 itself, so that your trait should be highly irritable. Otherwise, you are not able to select those plant. If it is yield, how you can select based on the individual single plant, based on that single plant performance, how you can judge the yield of that particular individual. Maybe in F2 it is in heterozygous condition, maybe there will be environmental effect. You can't able to differentiate for low irritable traits. If it is highly irritable, for example, disease or any single gene trait, so you can easily select from F2 itself. Okay, that's why it is highly useful when irritability is high. Okay. What are the merits? Gives maximum chances to select best plants, especially in early generation, as I told. Okay, helps in selecting transgressive segregants, plants better than parents. Okay, you can do in pedigree method. Faster than bulk. Okay, once you will see the bulk, you will get to know. Pedigree records helps understand inheritance of traits. How your traits are inheriting. Through the records, you can easily tell that. Okay, what are the demerits? Success depends heavily on the skill of the breeder. Okay, if you are not having that much skill, maybe you will miss the plants in F2 itself. If you are not selecting this particular plants, then you are missing the genotypes. Okay, that is a demerit. Natural selection does not play much role. Since you are the selection as a selection agent, there is no point of natural selection here. Early selection for yield is often not very effective, as I told. It is low irritable. Okay, early generation selection is not effective. It is too few. If too few plants are kept, the valuable genotypes may be lost, as I told. That's why your pedigree selection is having some demerits. Then modified pedigree bulk method. Okay, we'll also call it as a shuttle breeding, modified pedigree bulk method. Originally, it is developed by Norman Lee Borlaug, okay, in Simit, Mexico. When he is breeding the breeding for rust resistance or any the semi-dwarf wheat varieties, okay, there is some concept. In summer, that is in May to October, the seeds will be sown at Toluca region. It is high altitude, okay, and high rainfall area. And also it is high rust disease pressure. In winter, November to April, again after this generation, you will forward those material into winter in Obregon region. This is in the same that in Mexican, there is around the particular institute. Okay, there will be two different regions. These are low altitude near sea level and irrigated and hot desert climate. You can see this is short day, this is long day. Like that, they are settling the material generation after generation, one generation here, next generation here, then again come to this generation. Okay, what are the advantages of this particular shuttle breeding? They can select for long day, short day. Like this, they can do the photo insensitive genotypes. They can select for photo insensitive. There is no need to respond for short day or long day. If they are making photo insensitive, then they will be performing both in both the areas. Okay, and this is for high altitude, this is for low altitude. Okay, like that there will be so many advantages in case of shuttle breeding. Okay, who developed it? Normandy Borlaug in Simit. Okay, there are two regions, one is Toluca and Obregon. Okay, clear? कुछ जोश दिखा दो. 62 आ गया अभी 54. अरे व्यूअर्स आज तो पूरा कम हो गया. समझ आया ना? मास सिलेक्शन हो गया, प्योर लाइन हो गया, पेडिग्री हो गया. नेक्स्ट स्लाइड में एक इंपॉर्टेंट चीज है, बढ़िया है, आपको किसी को पता नहीं शायद. ओके, द हिस्ट्री ऑफ बासमती राइस. Why? Shinchan ask. Shinchan, why? Why? What's the meaning of why? Shinchan. There are two systems for pedigree record maintenance. अरे वो तो सारे छोड़ दो. There will be a different methods of numbering. There will be different numbers of, there is different ways to maintain the pedigree. It's again based on the different countries or maybe based on different breeders. They can maintain the pedigree record on their own way. It's not, don't worry about it. Once you become breeder, you can also maintain your pedigree on your own. ठीक है, मे बी मेरा PhD में बैकक्रॉस किया है, ठीक है, मे बी मेंटेन किया है मेरा हिसाब से, मतलब मुझे कैसे पसंद है ऐसे मैं मेंटेन किया है, नंबर तो मेंटेन किया है, ठीक है, वो तो उतना इम्पोर्टेंट नहीं है. वही मीन्स यस. अरे ठीक है, जेंजी है क्या आप लोग? No, it is not speed breeding. It is shuttle breeding. Speed breeding is very different again. Okay. Yes, you can use some off-season nurseries for speed breeding, but it is totally different, Ratnesh. In speed breeding, you can manipulate the time. You can manipulate the light and temperature so that you can grow 6 to 7 generation per year. In case of wheat, they have developed now by increasing the light period and temperature and humidity, by controlling these things, wheat can be grown 6 generation per year, 6 crops per year. For 2 months, there will be a crop season. Okay, that is speed breeding. Okay. Why means ऐसो सिंचन का ये पता है क्या आपको? जेंजी क्या है? आ गया नेटवर्क आ गया. नेटवर्क आ गया ना. क्लॉक इट मतलब विदिशा ये बोला. अच्छा है ना, पता नहीं क्या जेंजी लोग है ना आप लोग सारे. ठीक है, मुझे बाद में बताओ. This is about shuttle breeding. Okay, there is one beautiful example for pedigree breeding. ठीक है, We can also tell that the major achievement in case of basmati breeding, that is your Pusa basmati, which is released in 1989. The first, okay, the first semi dwarf basmati variety. Okay, Pusa basmati 1. It is developed by E.A. Siddiq and team in ICAR in New Delhi. World first semi-dwarf high-yielding basmati rice variety. Before that, there will be so many varieties. Okay, Pusa basmati 350, basmati 350, but those are larging susceptible, and also yield is very much poor. So that in export there will be so many problems. Okay, we are not able to export that much basmati to the outside countries. Okay, this is the one major achievement. Sir, please repeat the return once the network is gone now. कौन सा? Repeat rated one कुछ नहीं का है अभी बता रहा है बस. ठीक है, world first semi dwarf yielding basmati. This is the cross between Pusa 150 into kernel local. ठीक है, और Pusa 150 is transmitted the semi dwarf non-larging plant articles. Okay, this is the DG Ujan inserted variety Pusa 150. Okay, the semi dwarf gene, SD1 gene. Your kernel local, okay, it is elite basmati quality, extreme grain elongation, okay, and a rich aroma, that is your 2-acetyl 1-pyrroline. The aroma is responsible due to 2-acetyl 1-pyrroline in basmati. It is what they will do, they will cross between Pusa 150 and kernel local, and from that segregating material, okay, you are using pedigree method, they have released this Pusa basmati 1. Using classic pedigree method, breeders screened thousands of segregating plants from F2 to F6, selecting for the rigid dwarf statures and discarding the lines that lack the traditional cooking quality. This is one of the beautiful example for your pedigree method. There are so many different examples. Okay, again, छोड़ दो ये सारे. वो कब बल्क ब्रीडिंग से कौन सा वैरायटी डेवलप किया है, रिकरेंट सिलेक्शन से कौन सा डेवलप किया है, they will not ask you. They will not ask you. But for your knowledge, you should know the important achievements. Okay, for your knowledge sake, and maybe if they ask, they will only target some important, the notable achievements. ठीक है, वो कुछ छोटा छोटा रहता है ना, वो बीडीसी में लिखा है कुछ नरेंद्र राय, वो सा जंग मतलब ब्रासिका जंगसिया के क्या, वैसे कुछ कुछ छोटा छोटा एनपी सीरीज वाला वो लिखा है ना, वो सारे नहीं पूछ, ज्यादा आपका टाइम इन्वेस्ट मत करो उसमें. ठीक है, रतन सिंह, हाउ मेनी स्लाइड्स लेफ्ट सर? अरे भाई ज्यादा है, अभी भी सारे ब्रीडिंग मेथड्स खत्म करना है ना आज. फिर से आप लोग पूछेगा सर मैराथन. ठीक है, next is your bulk breeding. What we will do in bulk breeding? Again, we will start with hybridization. These are started with hybridization only. Okay, first we will create the variation, which is given by Swedish scientists Nilsson and Ehle in Swallow breeding station. In this method, after crossing selected parents, F2 and subsequent early generation are grown together in bulk without strict artificial selection. What we will do here, after the hybridization program, you will follow mass selection. Simply, if I want to say, okay, then whenever you will get interest to do something on that population, then you will start selection. Okay, थोड़ा आपको लगता है ना, अरे मैं 5-10 साल कुछ नहीं करना चाहिए इस पॉपुलेशन में, मैं ऐसे छोड़ना चाहिए, थोड़ा देख लेंगे क्या हो सकता है उसमें, उस समय आप बल्क ब्रीडिंग यूज कर सकते हैं. ठीक है, व्हाट यू विल डू, यू विल जस्ट क्रॉस एंड यू विल मेंटेन F2, F3, F4 सारे बल्क में मेंटेन करता है, मतलब सिलेक्शन भी कुछ नहीं करता है उधर, सारे बल्क में हार्वेस्ट करता है, नेक्स्ट जनरेशन बल्क, नेक्स्ट जनरेशन, ऐसे जाते रहता है. आपको पसंद लगेगा थोड़ा टाइम के बाद. ठीक है, उधर शुरू करेंगे आपको सिलेक्शन. वही है आपका सिंपल बल्क ब्रीडिंग. You will just bulk till F7, F10, F20 sometimes. Then whenever you want to do the selection, then you will start to do the breeding procedure. That is your bulk breeding. Simple. Okay, what are the applications of bulk breeding? Procedure तो आप देख सकते हैं इधर. You will cross P1 into P2, then you will get F1, then F3. You can see the number of plants you are maintaining may be similar, maybe same, or maybe they will increase. Okay, F2, F3, F4, F5. You will just bulk and plant in the commercial seed rate. Okay. Then after some F7, F5, F6, you will start to do the selection. Okay, you will select some 300 to 500 lines. Then you will, out of those again, you will select some 30, 50, then 10. Then at the end of the day, you will end up with any one good line. Okay, this is your bulk breeding method. What are the application? You can see isolation of homozygous line. Anyway, at the end of the day, as a self-funded crop plants, your product will be homozygous line. You can isolate homozygous line. Waiting for opportunity for selection. As I told, some disease epibiotics or any condition, maybe drought, maybe heat, or any disease will not come whenever you want. Okay, अरे भाई मैं इस सीजन में रस्ट होना चाहिए, वो भी ब्लैक रस्ट आना चाहिए मुझे, वो तो हम एक्सपेक्ट नहीं कर सकते हैं ना. वी हैव टू वेट फॉर दैट अपॉर्चुनिटी. ठीक है, फॉर द सिलेक्शन ऑफ प्लांट्स, फॉर स्क्रीनिंग. So that's why it is the best method. You can just bulk your material and you will go for next generation like that. Whenever your rust will appear after some generation, anyway your material also in homozygous condition at that particular time, your rust will come. You can select the plants whenever you want. Okay, you can waiting for the opportunity by using bulk method. You are doing bulking. Whenever you will get an opportunity, you will try to select and you will start the same pedigree method or any like pure line method. Okay, we will also call this as a mass pedigree method of Erlan. Since the beginning stage you will just follow the mass selection, that means you will just bulk it and move it. Then after when your opportunity will come, then you will start the pedigree selection. Okay, waiting for opportunity, as I told in the number of classes in live classes also. Okay, the two things you should remember in bulk breeding. One is your waiting. The application is for waiting for opportunity for selection. Second one is opportunity for natural selection. ये मतलब लेजी ब्रीडर्स रहते हैं कुछ लोग, उनके लिए अपॉर्चुनिटी फॉर नेचुरल सिलेक्शन. अरे भाई मैं कुछ नहीं करता है, मैं बस क्रॉसिंग कर दिया, P1 into P2, F1 बना दिया, मुझे मतलब मेरा मतलब वो मटेरियल का रिलेशनशिप कुछ नहीं है, मैं छोड़ दिया. कौन करेगा? आपका नेचर करेगा. ठीक है, you will go till F20, F15, F16, F17 and F18 like that. The nature will automatically select whichever the plants, okay, which are suitable for particular environment and whichever are fittest for the survival. Okay, the nature will select. We will call this as a evolutionary method of breeding by Sunesan. Okay, you will just bulk it and you will just grow, bulk, just grow. Okay, the inferior plants will automatically they will die and nature will select whichever the plants which are suitable for particular environment and particular soil and the different conditions, agroclimatic condition. Okay, everything is done by nature. This also major application of your bulk breeding. Okay, what are the merits? Natural or artificial disease outbreaks helps eliminate undesirable plants. Natural selection increases the superior genotypes in long-term bulks, as I told. Okay, the natural selection. Large population increases chance of getting transgressive segregants. If they ask which is the best breeding methods for exploit transgressive segregants, that is your bulk breeding. Yes, in pedigree also you can expect transgressive segregants, but the bulk breeding is the important one. In bulk breeding, you are not selecting it. You can see the 2,000, 3,000, 4,000 plants you are maintaining. So the opportunity to exploit those transgressive segregants, okay, to identify those transgressive segregants out of these 3,000 is very easy. Means maybe you will get the more chances. But in case of pedigree, yeah, you can expect translucent segregants, but you are selecting there, you are doing selection, so the chances is very less. Selection is done in later generation when plants are nearly homozygous, so it is more effective for low irritable traits like yield. Okay, suitable for small grain crops like the millets. Okay, for those crops you can go for bulk breeding, since you can go for space planting and you can easily maintain for the number of generation. Okay, there is no need to uh, go for space planting. It is very difficult to go the space planting in case of these kinds of smaller millets. Okay, you can just go for commercial rate and you can go till some 7th generation. Okay, it is best suited for high density crops like small grain crops. It improves adaptation to environment through the natural selection. ठीक है, ये तो आपका बल्क ब्रीडिंग की एवोल्यूशनरी में दो चीज आप याद में रखना है, वेटिंग फॉर अपॉर्चुनिटी फॉर सिलेक्शन, अपॉर्चुनिटी फॉर नेचुरल सिलेक्शन. नेक्स्ट इज एसएसडी. अरे ये तो सिंपल चीज, सिंगल सीड डिसेंट मेथड गिवन बाय गोल्डन इन मेज. Okay, what are the key features? First, you should know what is exactly the SSD method. You will cross P1 into P2, you will get F1. ठीक है, then you will get F2. Maybe around you will get 5000 F2 plants. मुझे अभी बताओ, there will be F2, then F3, F4, F like at the end of the day in SSD. Okay, when you will develop some lines, what is the outcome? The end of the day means after some generation, after some 5-10 years. Okay, what are the number of lines you can expect at the end? बताओ, जल्दी बताओ, कमेंट में लगा दो. I have given the number F2 numbers. I have given F2 number. Then I am asking, after using SSD method, what are the possible number of lines in the end? Yeah, real. Yes, Kavita, good. Recombinant inbred line. But I am asking the number of lines maybe you can expect at the end. जल्दी बताओ, जल्दी बताओ. अरे क्या हुआ? प्रिंसिपल तो पता है ना आपको एसएसडी का, 300 to 400. अरे सिंचन, क्या हुआ आपको? अरे भाई, एटलीस्ट you can guess. अरे भाई, मैं बताया P1 into P2 will cross, you will get F1, then from F1 to F2 you will get some 5000 F2 individuals. You will select single seed from each individual plant and you will grow in the next generation. Okay, you will go like that for F2, F3, F4, F5, then F6. Okay, you are started with 5000. Yeah, maybe you will lose some plants due to some natural calamities or inferior plants, but it should be around 5000 only. Okay, from each single F2 individual you will set single seed and you will bulk it. Okay, that's what I am asking. अभी बता रहा है बीट बाउंस. विदिशा, मैं वही पूछ रहा था ना. Yeah, as I told, maybe there will be some loss of some plants. There is more chance of loss of some plants due to some natural calamities. Yeah, but it should be around 5,000. Okay, maybe 4,500, 4,000 like that. हाँ, सिंचन लगा दिया 400, 500. क्या भाई, जीरो लगा रहा है क्या अभी? ऐसे मत करो सिंचन, ठीक है. Advances, what are the key features? Advances, maximum number of F2 plants up to F5, as I told. Only one random seed per plant. Okay, no selection in early generation. You are not doing any selection here. You are forwarding. You are just advancing the material. Main aim is to achieve homozygosity quickly. Okay, you can use your greenhouse facilities very efficiently. Since you are only 500 plants, 5,000 plants, then you are only selecting, only you are getting 5,000 seeds. You can easily, efficiently use polyhouse or off-season nurseries, greenhouse facilities, so that you can grow more number of generation per year. Selection starts after plants become nearly pure, F6 or F7. Okay, you can do the selection, you can release the variety. That is one kind. Also, you can, what are these 5,000 lines? These are recombinant inbred lines. That means from each F2 plants, you are ending up with the different recombinant inbred line. Okay, this is homozygous, but this will call it as a recombinant inbred line. To develop recombinant inbred lines, you can use single seed descent method. ठीक है, या modified method of bulk, ये बता सकते हैं सारे, ठीक है. And don't get confused for different modification methods. Okay, if they are asking any modification methods, they will definitely ask what exactly, what particularly it is. They will not ask you that modified. They will not confuse you guys. Modified bulk, pedigree, modified, different names are there. But what you should know, yeah, this is the modified method of bulk breeding. Okay, single seed descent method. ठीक है, next one is your backcross breeding. ठीक है, पेडिग्री हो गया, बल्क ब्रीडिंग हो गया, उसका मॉडिफिकेशन भी हो गया. ठीक है, अभी backcross breeding was first proposed by Erlen and Pope in 1922. It is used to replace one undesirable gene with the desirable gene, which is already established variety. For example, suitable recurrent parent SD3086 wheat variety, famous mega variety. Okay, but it got susceptible for rust in the recent years. Okay, then we should have some donor parent. Okay, any donor parent which is having rust resistance, but that variety is not suitable for cultivation, that is wild germplasm or any wild variety. Okay, but it is already established variety, your recurrent parent. Just we want to correct that particular disease resistance gene. Only we have to transfer the disease genes from donor parent to recurrent parent. The nature of trait should be highly irritable here. Highly irritable, like disease resistance. Adequate number of backcrosses, 4 to 5 backcrosses sometimes is needed. Okay, मेरा हैंड राइटिंग भी खराब हो रहा है क्या आप लोगों के साथ? हाँ, highly irritable. ठीक है, these are the different requirements: recurrent parent, donor parent, nature of trait, and adequate number of backcrosses. Now, how we will do the backcross program? ठीक है, you can see here, if you want to transfer dominant gene, okay, you are transferring the dominant gene. There will be different two procedure for dominance and recessive gene. You will see the difference. This is susceptible, that is your RR. You are susceptible. You want to transfer this capital RR, and everything, the other gene in the recurrent parent should be same as your recurrent parent. This is your objective. What you will do, first you will cross, then you will get F1, that is capital R and small r. Your capital R is responsible for disease resistance. Then you will cross with small r again. That means you are backcrossing with the recurrent parent. Your F1 you are backcrossing with the recurrent parent. Then whatever you will get, this is capital R, small r. Then you will backcross with this. This is F1. Then whatever you will get, that generation we will call it as a BC1 F1. What you will get? BC1 F1. You will get capital R, small r, and small r, small r. Anyway, your small r is susceptible. Okay, you will screen for disease. You will inoculate disease. Then you will screen small r will die. Okay, your BC1 will get small r, and capital R, small r when you will cross with small r. Okay, but this small r will not survive when there is a disease. Okay, since your R will responsible for disease resistance. Then again this RR will backcross to your small r. Like this you will do BC1 F1, then BC2 F1, like this BC3 F1. Like this you will go for some BC3, BC4. Then after some generation, okay, you will try to self. Okay, after some generation, anyway, you will get small r, capital R, small r. Then you will self this thing, seeds, capital R, small r, capital R, small r, small r. Anyway, you will reject this. Then you will grow this. Okay, which are the lines which are not segregating? It will segregate na capital R, small r, but it will not segregate. So that you will reject these lines. We will only forward these. Okay. Now the ultimately your resistant cultivar, okay, is having capital RR gene in the background of your recurrent parent. In the background of your HD 3086, but it is having dominant gene. What you will do? You will just backcross some of 4 or 5 backcrosses. Then you will go for selfing. After that, you will select non-segregating generation. This is your simple method of transfer of dominant gene in backcross breeding. But in case of transferring recessive gene, okay, what you will do? Your susceptible parent is having capital R. Your resistance is small r. Okay, your recessive gene is responsible for dominant or disease resistance. But your recurrent parent having capital R. First, what you will do? You will cross them. You will get capital R, small r. Okay. Anyway, it is susceptible again. Okay, there is a dominant gene here. If you want your recessive, you want resistance, then your recessive gene should be in homozygous condition. What it will do here? You will backcross to particular recurrent parent, or you can also go for selfing. There are 2 different choice you have. If you go for selfing, then you will get capital R, small r. One chance. If you go for backcrossing, there also you will get capital R, small r. Next, you will go for selfing. You can directly go for selfing, or you can go for backcross, one more backcross, and go for selfing. After selfing again, same, you will get like this. Selfing, after selfing, you will get 3 genotypes. In that particular case, what you will do? You can easily discard these, since these are susceptible. Your resistance is due to recessive gene. Okay. You will only select small r. Then you will go for backcrossing again. Then if you cross this, then you will get heterozygous dominant. Again, you are not able to screen here. Okay, this is due to capital R, small r. So your resistance is due to recessive gene, so it should be smaller condition. Okay, to get that, you will again go for back cross, capital R, small r and small r, then you should self. That's what this method will tell. After every two back crosses, you need to be followed by selfing so that progeny with homozygous recessive genotype can be identified and backcrossed to the recurrent parent. In this particular case, if when you are transferring dominant gene, okay, whatever you are getting after backcross, it is resistance, so that you can easily, you can see here, after each backcross, okay, you will get small r and capital R smaller. You can reject small r and you can backcross the capital R smaller to the recurrent parent. But in this case, it's not like that. Okay, your capital R small r in after every backcross. Okay, you can see capital R smaller, capital R smaller. After every backcross, it is capital R smaller, but your resistance is due to recessive gene. You want to expose that, so that you should go for selfing. After every two backcross, you can go for one selfing. Okay, if you go for selfing, so that you can express that particular, you can identify the resistance gene due to recessive, so that you can discard these two. There is a need of selfing. What you should remember here, there should be a selfing in case of when you are transferring recessive gene after every two backcrosses, so that you can expose that particular homozygous recessive condition. Okay. Nanda Kumar KR, क्या चाहिए नंदा कुमार? What energy? Love you sir. अरे thank you because fan of your energy sir. समझ रहे हैं ना नंदा कुमार? Okay. Can you tell me why we need for selfing in transferring recessive gene? नंदा कुमार बताओ. Why we need of selfing in case of transferring recessive gene? अरे repeated sir, repeat कर रहा है ना, बताओ अभी. As I told, okay, we will go for selfing after every two backcrosses. Okay, when we are transferring recessive gene. Okay, what's the need of that selfing? बताओ, कोई बताओ. नंदा कुमार को पता नहीं शायद. टाइपिंग सर 1 मिनट. ठीक है, और कोई बता सकता है क्या? अभी मैं क्लियरली बता दिया. बताओ ना आंसर. टू आइडेंटिफाई रिसेसिव जीन सर or homozygosity, to expose recessive homozygous. That's what I wanted answer. Okay, Lucky Patel. Yes. To expose the recessive gene. Otherwise, it is in case of dominant, when you are transferring dominant gene, you can easily identify. Okay, your resistance due to dominance. But in case of this particular case, to expose that condition, you should need that selfing, yes, to get homozygous recessive. Yeah. Ask one more question. अभी इसको आप आंसर सही से नहीं बता रहे, फिर से क्वेश्चन चाहिए क्या नंदा? मुझे पता है नंदा कौन है. नंदा हम मेरा जूनियर है शायद. ठीक है, देखो वो एमएससी कर रहा है, आप लोग तो ऐसे अच्छे से आंसर दे रहा है, उसको कंफ्यूजन हो रहा है. ठीक है, एप्लीकेशन. What are the application of backcross breeding? Intervarietal transfer, simply inherited traits. Transfer of quantitative characters, you can also go for transfer of quantitative characters sometimes. Okay, interspecific transfer between species, you can just backcross. Transfer of cytoplasm. Okay, this is your female parent. Okay, when your sterile cytoplasm. Okay, and you want to transfer your cytoplasm. This is small a, for example, it is fertile. Okay, like this, you will get F1, then you will backcross this particular genotype, continuous backcrossing. Okay, so that you can transfer this sterile cytoplasm into another line. Okay, this is due to backcrossing. Recovery of transcriptive segregants again. In this case also, in backcross also, okay, maybe in backcross after 3-4 backcrosses, you can go for transcriptive segregants. Okay, and also the combination breeding again. Okay, when you want to merge your yield with disease resistance, okay, you can go for combination breeding also, transcriptive breeding also. But the thing you should remember here, the best method for transcriptive segregants, the more chances will be in bulk breeding. Okay, and the good breeding method, okay, the practically your pedigree will be for combination breeding. To changes any defects or any corrections of particular single inherited genes, you can go for backcross breeding. Okay, production of near isogenic lines. Near isogenic lines are nothing but when you do continuous backcrossing to particular gene. Okay, now what we have developed here? First one is your SD3086, the original line. Then after development, again it is SD3086 only. But you have introduced one resistance gene for this particular variety. Both are isogenic, nearly isogenic. Okay, when we will call isogenic, those lines only differentiate for any single loci. Okay, why we will call near isogenic lines? It is always, not always, they will be difference for particular single gene. There should be around. Okay, when you are transferring particular chromosome location, this is your gene. Okay, when you are transferring this gene, okay, this is your SD3086, original SD3086. When you are transferring this particular gene to this particular line, there is a flanking regions also will come sometimes. That's why we'll call it as a nearly isogenic, not completely isogenic lines. Okay, nearly isogenic lines due to that flanking region. This is about your backcross breeding. Clear? I am PhD sir, not MSc. Nanda, TK bye. You are my favorite senior sir always. Thank you. TK. नंदा तो पीजीआर का साइंटिस्ट है, ठीक है बच्चों, वो साइंटिस्ट बन रहा है. Different only, yes sir. Okay, then multi-line breeding. First suggested in oats by Jensen in 1952, theoretically, but the application, okay, application of that particular multi-line concept given by Borlaug and team. Okay, mainly used in self-planted crops like wheat, oats, soybean, groundnut. Okay, maybe you have heard these lines: KSML 3, KSML 11. What about KSML? Full form? मैं क्लास में बताया था शायद. गुरु शशांक, एक फिर से एक बार वो लिंक शेयर कर देना सारे सब ग्रुप में. सेकंड पार्ट शुरू होने वाला है इसलिए बताओ, सारे बताओ. एक कदम करके क्रॉस प्लांटेड शुरू करेंगे, क्रॉस प्लांटेड क्रॉप्स. ठीक है, KSML कल्याण सोना मल्टीलाइन गुड कविता, KSML 3 and KSML 11. These are actually the mixture of 8 multilines. 8 multilines. Okay, what we will do here? We will just mix the pure line varieties. Okay, one of the pure line variety is resistance for one of the race of rust, like that. One of all multilines, okay, all the pure lines within the multilines are having some one resistance genes. So those are resistance for each races of particular rust disease. We will mix those pure lines and we will release for the cultivation. This is the concept of multi-line breeding, which is originally given by Jensen but practically applied by Norman E. Borlaug in CIMIT for the wheat cultivar development. As I told, there is a two approach here. Clean crop approach, dirty crop approach, given by Marshall. Okay, again this is the concept of multi-line only. But what will happen in case of clean crop approach and dirty crop approach? In clean crop approach, the each components of multi-lines, okay, each lines within the multi-line are having resistance gene for all the prevalent races. Okay, as I told, if for example, you have developed any multi-line which is having mixture of 8 pure lines, that particular pure line, each pure line having resistance genes for all the rust disease, all the races of rust disease. Okay, it is completely free from diseases. If any disease attacks, then completely it is free. But if any particular race got evolved and if it attacks, then complete crop failure. Okay, at that particular time it is having resistance genes for all the races. All lines are having resistance genes. Okay, but in case of dirty crop approach, none of the lines are having resistance gene for all the lines. Okay, what it will do? It will reduce the load of infection. Okay, if any new race evolves also, okay, there is not that much crop loss in case of dirty crop approach. Okay, there will be some less yield per se, but it will not allow to evolve the races, or also it will not that much yield loss in case of dirty crop approach. But clean crop approach, yes, it is having resistance gene, all the lines, all the pure lines within the multi lines are resistance for all the races. But if anything happens out of the box, then it is in the danger. Okay, this is the two approaches. No selection pressure in dirty line approach, selection pressure in clean crop approach. Selection pressure, selection pressure क्यों बता रहा है पता नहीं मुझे. Sinchan, repeat sir these both. Kavita, this is simple. In clean crop approach, the multi lines, for example, if you are having 8 lines, 8 pure lines in your multi line, all the lines, okay, each line in the multi line are having resistance gene for all the prevalent races. Okay, for example, in case of bacterial blight BLB, okay, there is a XA5, XA13, XA21 gene. Okay, these 3 genes are present in all 8 lines. That is your clean crop approach. In case of dirty crop approach, some are having XA5, some are having XA13, some lines. Okay, some lines are having XA21, some lines are XA5 and XA13, like this. None of the lines are resistance for all the particular races. समझ रहा है ना अभी? कविता बताओ, जल्दी बताओ कमेंट में. Okay, now breeding for self-managed croplands, now it is over. Now whatever the important things maybe they can ask for your exam, now just now we have completed. Now this is time for breeding methods for cross-pollinated crop plants. The one thing you should remember here, whenever you are talking about breeding methods for self-managed crop plants, you will always target individuals. Okay, you will try to select individuals. Okay, selection is very important. But in case of cross-planted crop plants, okay, the population improvement is the main objective. You are not worried about single individual there. Since it is heterogeneous materials, heterogeneous mixture, okay, you will try to develop population. You will try to improve the population, okay, to accumulate favorable alleles in the population. That is your objective here. But there, you will try to isolate individual line in case of self-planted crop plants. Okay, this is the major difference. Okay, got it, sir. Got it. Yes. What breeders aims to increase the frequency of desirable genes in the population. Since okay, this is a group, as a group, the population is important here, so that we will try to accumulate the frequency of the desirable genes, create new useful genotypes, maintain genetic variability for future improvement. These are the main objectives or aims we can tell in case of cross-planted crop plants. Okay. Accumulation of favorable alleles in the population. This is the main objective of your breeding for cross-planted crop plants. So again, mass selection, the simple breeding method. Okay, whether it may be self or whether it may be cross-planted. Okay, you will just go to the field. Okay, original population. You will select the superior plants based on the phenotype. Okay, then there will be self-pollination. Yours whatever the seeds you will select, those are self-pollinated seeds. But ये तो इधर ऐसे नहीं है. Mass selection, sorry, cross-planted crops में you can't go for selfing here. Okay, you will select some superior plants and whatever the seeds set on that particular self-planted, sorry, cross-planted crop plant are due to open pollination. Open pollinated seeds from the selected plants are harvested in the bulk. Then you will grow bulk seeds in the next generation. Like that, you will just select the plants, you will bulk the open pollinated seeds, cross-planted seeds, then you will go for next generation. Then you will go for at the end of some 2 to 3 years, you will go for yield trial. Then you will try to see the best variety. This is the mass selection. Okay. Next, modification of mass selection, mostly used in cross-planted crops. You know now the defects, the problems with the mass selection. No control over the population. As I told, the seeds which are setting on the selected plants are due to open pollination. The pollen is coming from somewhere else. Don't know, maybe it is coming from superior plants or inferior plants. There is no control over the population in the mass selection. Selection based only on the phenotype. We are not doing progeny selection also here. Okay. To overcome those defects, you can do one that there are other things. You can do those things. The rejection of inferior plants. Okay, in the population, okay, when you are selecting the superior plants, you can also try to remove those inferior plants so that the pollen should not come from that particular plants. Or okay, you can use composite pollen. That means you can collect the pollens from the open pollinated, sorry, the superior plants, and you can use those pollens for the crossing with that particular selected plants. You can also go for that kind of method. Otherwise, you can go for stratification of field, that is grid method given by Gardner. In case of mass selection field, okay, the phenotype, it is highly affected by environment. So you can't only rely on that phenotype. So you will just, what you will do, you will just try to separate the fields. Okay, we will just try to separate the fields on like the grids, different grids. Okay, you will just try to select the plants from that particular grid. Okay, like you will just divide the field into some 10, 12 small plots. Then you will try to select the plants from that particular plots. Then you will just compare, okay, among the different plots. Okay, this is the one of the method, the grid method. Okay, to overcome some defects. Next, selection schemes with progeny test. Okay. Without progeny selection is your mass selection. Okay, in case of cross-planted croplands, without progeny selection is your mass selection. With progeny selection, year to row method, your recurrent selection also method of progeny selection. How? I will tell you. Okay, now the progeny selection, the year to row methods is given by Hopkins in Maze. Okay, year to row method. The original population, you will go to the field and you will select some 20 to 30 superior plants and you will grow those 20 to 30 plants in year to row method. You will just select that particular cob and you will grow in the next generation as a progeny rows. So you can evaluate those progenies, okay, based on the particular phenotype. You can just see which are the superior plants, superior progeny rows. Small individual plant progenies are grown in the selected plants. Superior progenies are identified. Phenotypically, superior plants selected from the superior progenies. These are your superior progenies. Out of these superior progenies, you will select the best plants in that particular progeny row. Then you will grow for next generation. It is one of the progeny selection method. Okay. Next, in case of second scheme, okay, it is a cycle. After that, you can just again start with the second cycle like that. And at the end of that, you can go for yield trial and you can release the variety. Another scheme, in second scheme, what you will do, you will select some plants out of 100 plants in the field. You will select some 10 to 15 plants. Then you will just, in the second year, what you will do? You will just grow as a progeny rows of some of the self-food seeds. Then some self-food seeds, okay, you will grow in the next generation. What you will do here? You will just try to see the superior progenies first. Then based on the superior progenies, you will only grow those superior progenies in the next generation. Okay, only those pollens will pollinate in this particular case. Okay, what you will do, you will just follow the same first scheme in the first step. Okay, the remaining seeds from those plants selected in the first year that produce the superior progenies are bulked to rise the next generation. The bulk seed are planted, plant allowed to open pollinated. Okay, here you will, the thing is, in the second case, you will go to the field, you will select some 100 lines, 100 plants. Okay, you will collect the 100 plant seeds. Some of the seeds you will grow in the progeny rows. You will evaluate those progenies. The whichever progenies are giving good superior performance, those progenies, the remaining seeds of those progenies, you will sow in the next generation and you will try to leave for open pollination, so that you can avoid those inferior progenies. Here, in case of inferior progenies, okay, here what they will do, in the progenies rows, your inferior progenies will also maybe pollinate the superior progenies. So, okay, you can avoid that. Okay, based on the superior progenies, you will grow those in the next generation and you will leave for open pollination. Okay, that is the case in this particular progeny test. Okay. Now, modified year to row method, it is given by Lundquist in 1970s, in 1964. The thing is, here the 2 points you should remember. Progenies will be tested in replicated yield trial. In case of year to row method, there is no replicated yield trials. Okay. In this particular case, modified year to row method, you will go for replication yield trial and pollen source also controlled. Okay, you can control over the pollen source. You can choose whichever the male parents, whichever the pollen source, okay, which are superior or inferior. You can control that pollen source. Selection cycle completed in 1 year. Okay, you will just select the plants in the same year itself, and you will also control the pollen source, and you will also go for replicated yield trial. These are the 3 things you should remember in case of modified year to row method. Okay. Okay, this is all about year to row selection. Okay, next will be your recurrent selection. You can see here, as I told, your year to row method also progeny selection method. Okay, your recurrent selection also progeny selection method. Here we are doing the evaluation of progenies. You are doing the evaluation of progenies from the selected individuals. Okay, but what is the method you are following for the progenies? These are open pollinated seeds. You will select the seed, you will select the plants, but they are open pollination, they are open pollinated seeds. Then you will use those progenies for the evolution. But in case of recurrent selection, there is some restrictions. Okay, whatever progenies you will evaluate, okay, those are three different things. In simple recurrent selection, the progenies you will evaluate, the progenies from the selected plants you will evaluate are the self-food seeds in simple recurrent selection. Okay. In case of recurrent selection for GCA, the progenies are crossed seeds with the open pollinated variety. In case of recurrent selection for SCA, the progenies you will evaluate which are crossed with inbreed or any narrow genetic base tester. ठीक है, समझ रहे हैं ना अभी? What I am telling is, okay, you can see here, the progenies, your recurrent selection is also progeny selection. But what the thing you should remember here, okay, in case of year to row method, the progenies which you are going to evaluate, okay, you will evaluate progenies to select the superior individuals. Your progenies are giving good performance, then you will select only those progenies, only those individuals, right? The progenies what you will evaluate in case of year to row selections are open pollinated progenies. But in case of recurrent selection, simple recurrent selection, okay, your progenies are self-food seeds. In case of recurrent selection for GCA, okay, your progenies are crossed seeds with open pollinated variety as a tester. Your recurrent selection for SCA, okay, crossed with any inbreed or hybrid, that is your broad narrow genetic base. Okay, this is the scenario. Okay, it's a simple thing you should know, so that you can easily understand what exactly the recurrent selection is. Again, recurrent selection is nothing but the accumulation of favorable alleles in the population. It is a method of improvement, the population improvement. Okay, what you will do? Selection, intercross, and selection. Selection after generation. You can see the cycle is like, first you will select the plants, you will evaluate the progenies with the tester. Whichever progenies are giving the best performance, you will allow those plants to intermate among themselves. ठीक है? उसके बाद again फिर से second selection cycle, you will select and you will cross with tester, whichever giving good performance, You will allow them to intercross among themselves. The simple thing. But which are the progenies you are going to evaluate? Okay, and which is the tester you are using? It will only depends. Proposed by Ace and Garber independently by East and Jones, further developed by Hull in 1945. What are the important features? As I told, it is a type of progeny selection. Differs from simple progeny selection because progenies are obtained differently. In case of paired row selection, your progeny is from the open planted variety, open planted seeds, but here it's not the case. Selected lines are intercrossed, but in near row selection, you will just open for open pollination. But here selected lines are you are making intercross among themselves. Suitable for quantitative traits, for example, yield, best for highly irritable characters also. Inbreeding can be means you in this particular breeding method, you are minimize the inbreeding through the random mating for one generation or avoiding selection from the closely related crosses. Okay, now we will see each one by one how we will do the simple recurrent selection. Okay, as I told, the concept you should understand first. Okay, the original population. What you will do, you will go to the field, you will select superior plant progenies. Okay, out of some 1,000, you will just try to select some 100 good individuals. Okay, selected plants are self-pollinated. Okay, you will just self-pollinate them. In case of paired row selection, you will just select and you will just collect the cobs. In case of maize, you will just collect the cobs. Those are open pollinated cobs. Okay, but here you will make the intentional selfing of that selected plants. Okay, then self-food seeds are harvested separately. Then self-food seeds are evaluated. Okay, you can see you will evaluate those self-food seeds in the next generation. Okay, individual plant progenies are planted. All possible intercrosses are made. The best progenies which are having the good performance, you will make intercross among themselves. Okay, among themselves, equal amount of seeds from all the intercrosses are composited. Okay, the self-food seeds you will evaluate and you will intercross among themselves. This is original selection cycle. Then again, you will select, you will self the seeds, you will sow in the crossing block in the next year. Okay, you will cross among the best superior, you will intercross the superior progenies. This is the first recurrent selection cycle. Like that, you will just go one after another. Okay, so that you can make the improvement in the population. Okay, the simple selfing, self-food seeds, selfing seeds, self-food seeds are your progenies. Okay, then after that you will make the intercross of best progenies. But in case of recurrent selection for GCA, it is off-SIP progeny test. We can also tell. Okay, off-SIP is nothing but, okay, you are crossing your original population with the open planted variety. Your open planted variety is a mixture of different genetic constitution. Okay, whatever. Is it clear now? अरे भाई आज तो व्यूअर्स भी कम है, नेटवर्क भी सही नहीं है, अब आपका जोश भी कम है. ऐसे है क्या? अभी क्लियर है ना? बताओ क्लियर है ना अभी वॉइस और वीडियो दोनों? Okay. Recurrent selection for GCA is direct outgrowth of early testing suggested by Jennings. Okay, in 19 35. What we will do here? It is like an early testing for the inbreds. Okay, in case of development of inbreds, we will test for GCA, how they are combining with open planted variety. If they are having good combining ability with open planted variety, that means they will combine well with each other, with other inbreds. Okay, what we will do, as I told, the progenies, okay, the test class progenies, what you are going to evaluate are crossed seeds with the open planted variety. You will go to the field, you will select some 30 or 40 plants. Each plant you will cross with the open planted variety. Okay, and also you will self those seeds. You will self those seeds. The crossed seeds you will evaluate first, the progenies. The whichever individuals, okay, what you will do? For example, 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, you will cross with OPV. Okay, you will get progenies, right? For each individual, you will cross with OPV, and also you will self. You will, what you will do first? You will evaluate these particular crossed seeds in the field. You will just self seeds, you will kept separate. You will evaluate these crossed seeds. Whichever lines, okay, whichever lines among these 9 are giving good performance, okay, in the test class progenies with the crossed with OPV, okay, you will only use, for example, okay, these 1, 5, 8, these 3 progenies here will give good performance. We will only sow those 1, 5, 8 seeds progenies in the crossing block, and you will make the possible intercrosses. You can see self-food seeds from the plants producing superior test class planted in a crossing block. All possible intercrosses are made. Equal amount of seeds from all the intercrosses are composited. Okay, that is your first cycle. The same cycle will be your first recurrent selection cycle. This is the original selection cycle. Then again you will do the same. Again you will use this tester, and you will use the progenies. Okay, then based on the progenies, using self-food seeds, you will go for intercross among themselves. ठीक है, सेम ही करेगा अभी. मैं एक अभी क्वेश्चन पूछ रहा है, सब रेडी हो जाओ अभी. ठीक है, गिफ्ट तो पक्का मिलेगा. कौन बता देगा? मैं देखता हूं. अरे भाई मैं बता रहा हूं. ठीक है, ये क्रॉस कर रहा है ओपीवी से. For example, this is the first individual. I am crossing with OPV to get test class progenies. Okay, to evaluate in the next year. I am doing selfing also. अरे कैसे हो सकता है भाई दोनों एक ही प्लांट में, क्रॉसिंग और सेल्फिंग? बताओ, कोई आंसर बताओ इसको अभी. I am telling, I am doing selfing also to keep the seed separately. Okay, then I am doing this also. Lucky Patel, tester as female. Okay, I will use tester as female. Then बताओ, selected plant as a male. Selected plant as a male. Okay, अरे पूरा बताओ. यस लकी पटेल, आप सही बता रहा है. पूरा बताओ आंसर. क्रॉस पॉलिनेट. अरे एक एक वर्ड डाल रहे हैं, एक वर्ड भेज रहा है. What you will do? For example, if you take as a maize, whenever it is possible in monoecious crops. Okay, Selected plant is pollen donor for crossing. It is also self beside. Yeah, Sajan. Okay. The thing is, it will be useful in monoecious crops like maize. Okay, the specific recurrent selection is designed for maize crop. Okay, for example. So maize having different male and female flowers. Okay, you will take pollens from this particular plant and they will cross with the OPV. So these seeds will be crossed seeds. Then the male pollens, okay, you will just pollinate with the cob. So you will get an self seeds. ठीक है? लकी पटेल, आप हमारा कोर्स का स्टूडेंट है क्या? साजन, दोनों बताओ, दोनों हमारा कोर्स का स्टूडेंट्स है क्या? ठीक है, दो आप मतलब हमारा कोर्स का स्टूडेंट हो तो आपको मेरा नंबर तो पता है, ठीक है, एक मैसेज डाल दो एड्रेस के साथ। तो साजन आप तो नेमानी को ऑलरेडी गिफ्ट तो भेज दिया है फिर से नहीं मिलेगा ठीक है, लकी पटेल आपको मिलेगा, जस्ट एटलीस्ट यू हैव ट्राइड। ठीक है, आपको एड्रेस के साथ में एक मैसेज डाल दो मुझे। ठीक है, so this is about your recurrent selection for GCA. So what you are doing here, you are improving, okay, the base population for the general combining ability. Okay, at the end of the day, whatever the population, okay, whatever the end product you are getting are good inbreeds. Okay, the good inbreeds which are having good general combining ability. So those inbreeds you can use for yes, whatever the end product of this particular population improvement, okay, what those inbreeds you can use for, batao? बताओ यार, मैराथन चाहिए आपको, अब कोई नहीं सही से रेस्पॉन्ड कर रहा है। हाँ, वसुप्रदा SCA, अरे भाई, I am asking the end product, the inbreeds which you are developed through the recurrent selection for GCA will be helpful in for which purpose? The inbreeds which are having good GCA, okay, are the source material for hybrid. For hybrid, मधु एगड़े hybrid बता रहा है रे, वही मधु, it's not hybrid. The inbreeds which are developed from SCA will be used for hybrid. Okay, अरे मैं तो एक्सपेक्ट नहीं किया था hybrid. Then what you will do with yes Kavita, for synthetic variety, the source material for synthetic variety, what about source material? Okay, the inbreeds which are having good GCA, right? Okay, the inbreeds which are having good SCA, okay, we will use for hybrid development, which are having specific combining ability, which are giving good performance with the specific inbreeds. Okay, those are hybrids, we will use for hybrids. But you are yes Madhu, good. Now you guys are throwing synthetics. Okay, what about SCA? It is given by Al. The simple thing, okay, your inbreeds or any genotypes, okay, 1 2 3 4 5 6 7 8 9 10 11, okay, you will cross with your tester will be like here any inbreed, okay, any inbreed or hybrid. Okay, this is narrow genetic base. Your OPV in GCA, that is broad genetic base. Narrow genetic base. What you will do? You will just cross. Okay, you will test cross progenies. You will also go for selfing of particular individuals. Okay, whichever the progenies we are having good SCA, okay, like 10, 9, and 2, which are having giving good performance with the test class progenies. You will self those seeds, okay, then you will grow those selfed seeds and you will make intercross among themselves, and you will take equal amount of seeds from intercross and you will bulk it. This is original cycle. Then you will go for first recurrent selection cycle, like that, second, third, like that. Whenever you want to satisfy, whenever you will satisfy, then you will try to select some good inbreds for hybrid development. Okay? Yeah, well, the thing is, in inbred in case of hybrid development, first you will take the inbreds which are having good GCA. I will agree. Okay, the inbreds having good GCA, first you will take some 100 inbred lines, okay, which are having good GCA. Then you will make the crosses among themselves. Then you will see first SCA among themselves. Among those 100, if you are using some 10 inbred lines, there are possible number of crosses will be n of n minus 1 by 2 direct crosses. Okay, out of those crosses, you will get some good. There will be some 3-4 crosses which are having good specific combining ability. You will release those as inbreds. This is the procedure. I will agree that yes, you will use the GCA inbreds having good GCA for hybrid development also. But the source material for synthetic development will be your inbreds with the GCA. Okay. Okay, this is the major protocol schematic representation of for recurrent selection for SCA. In this case, the end material, whatever you will get, are having good SCA. Okay, so that you can push those inbreds for the hybrid development. Okay. Reciprocal recurrent selection. Okay, the thing is here, the testers are for original population A, the tester will be population B. The population B, the tester will be population A. Okay, you will evaluate the progenies, test class progenies in the both the cases, and you will also go for self-food seeds. Okay, then you will intercross those progenies which are having good performance in the self-food seeds of that particular progenies. You will make the intercross and you will use those seeds in the composite. You will make the composite of equal quantity of the seeds. Then you will go for next generation. The simple difference is here, the testers will be complementary to one another, so that you are improving two populations, okay, simultaneously. It is proposed by Comstock, Robinson, and Harvey. Actually, the thing is, this is the best or efficient method of recurrent selection. Since you can see this original population, you are using tester for this particular population, so that okay, it will exploit both GCA and SCA. Okay, it will act as narrow genetic base and broad genetic base simultaneously, so that you can exploit, okay, both specific combining ability, general combining ability simultaneously, and also you can simultaneously improve the two population at a time. Okay, that's why it is one of the efficient recurrent selection method. Why a good tester should not have high yield? अरे भाई, ये तो सिंपल चीज है, कोई बता सकते हैं क्या? Why we will use the testers which are having not good yield? उसका नाम टेस्टर, ठीक है सिंचन, उसका नाम ही टेस्टर है, वो यूज कर रहा है टेस्ट करने के लिए. आपका इनब्रिड्स है ना, आपका ओरिजिनल पॉपुलेशन है ना, उसका परफॉर्मेंस, उसका पोटेंशियल बाहर लेने के लिए. ठीक है, इफ इट इज योर टेस्टर itself is an high yielder, then how we will conclude that, okay, whatever you are getting the progenies, okay, whatever you are getting the progenies are due to your tester or your inbred. How we will get to know? If it is low yield, okay, then if you are getting any progenies which are having high yield, that you can easily tell that the potential is due to your selected individuals. ठीक है, समझ गया ना सिंचन? सिंचन आप भी एक मैसेज डाल दो मेरा नंबर से, आपको भी मेरा नंबर तो मिलेगा ना ग्रुप में, आप भी एक मैसेज डालना है मुझे, ठीक है सिंचन, भूलना मत। Okay, reciprocal recurrent selection. If I want to conclude this particular recurrent selection in a single table, okay, you can see here gene action incomplete dominance, where there is an incomplete dominance. Okay, your reciprocal recurrent selection is always best method. Okay, compared to GCA. Okay, additive effects are important. मतलब your reciprocal recurrent selection equally effective when it is with the recurrent selection for GCA. As I told, recurrent selection for GCA is offsheep progenies, so you can evaluate the additive gene action there. For example, your particular selected inbreed, you are crossing with OPV, so that you will get number of progenies here of the same particular individual, particular inbreed. Okay, when you will cross with OPV, your OPV will produce the each seed which will set on the OPV. This is your cob, na? Cob contains so many number of seeds. Okay, what you will do? You will take the male gametes. Okay, your male gametes are, it is like homozygous, or you can also tell that it is completely similar genotype. Okay, so that you will cross with OPV, but OPV will produce the number of different female gametes. So each seed in the cob is very different. So that in the progenies, okay, whatever you will get the different genetic constitution progeny, so that you can easily estimate the additive gene action of that particular inbreed. Okay, how additive, that's what the general combining ability. Okay, how it is combining with the different gametes of OPV. That's why it will tell the additive effect, your recurrent selection for GCA. Complete dominance, when there is a complete dominance, okay, all your three will be equally effective. But when there is an over dominance, as I told, your dominance will give the heterosis. Okay, that means there is a specific combination. Your reciprocal recurrent selection will be equally effective for recurrent selection for SCA when there is an over dominance. Okay, since in over dominance, what you will do in case of recurrent selection for SCA, what you will do? Will cross with your selected individuals with the any inbreed or hybrid. These are homogeneous material. All will produce same types of gametes. So whatever individuals the progenies you are getting, okay, they are similar, so that you can exploit. You can estimate the heterotic value. Okay, so when there is an over dominance, you can easily exploit, okay, this particular recurrent selection for SCA. Practically, okay, when there is an epistatis, practically there is a presence of epistatis also, so that your reciprocal recurrent selection will be effective. It will capture additive and non-additive both. As I told, it will act as both narrow and broad genetic based tester. Okay, and also it will be simultaneous improvement of two population. This is about your recurrent selection. Then हो गया आपका progeny selection हो गया, population improvement हो गया. Okay, now we are into the hybrid. Okay, hybrid cultivar is the F1 generation produced by crossing two different parents in a planned way. If I ask you what is the difference between F1 and hybrid, बताओ कोई? What is the difference between F1 and hybrid? कोई बता सकते हैं क्या? लगा दो कमेंट में. What's the difference between F1 and for hybrid parents must be genetically different. All F1s are hybrid, but not all hybrids are F1. Good, Nemani. That's what the correct difference we can tell. F1 between two same of different parents. Same or different parents. Yes. F1 is the particularly first offspring generation of a cross, but hybrid between two different parents. The thing is, as Nemani told, okay, all F1 are hybrid. Okay, hybrid is nothing but the cross between any two individuals. Okay, but all hybrids are not F1s. For example, double cross hybrid. Okay, double cross hybrid is an hybrid. Okay, we will call it as a hybrid, but it is not F1. It is not F1, right? Like that. For example, any cross between the any parents, as someone told, okay, Kavita, I think, any parent, any cross, this is the first filial generation F1 is. Okay, but if you want to call it as an hybrid, it should be in a planned way, the planned cross, and also it should show some vigor, hybrid vigor or heterosis. Then only we can call your F1 as an hybrid. Okay. So hybrid vigor, it is nothing but your heterosis. Okay, the term is given by Shell. If you want to take an formula, okay, hybrid vigor, the F1 over the better parent, any parent, okay, the performance of an F1, the superiority of an F1 over the parents, then only you can call it as an hybrid vigor or heterosis. The F1 should have some superiority over the parents. But in case of your F1, it is not like that. If you cross tall into dwarf, you will get F1 as a tall. It not may be superior, or it may be sometime it may be mid parent value. Okay, if F1, if it is incomplete dominance, your F1 will be mid parent. Okay, that's your F1. But in case of hybrid vigor, if you want to tell you are telling any term heterosis, then it should be your F1 is superior over the parents. Then only you can call it as an hybrid vigor or hybrid. Okay. What about inbreeding depression? The loss of vigor and fertility. Okay, the loss of vigor and fertility during selfing. If you go for selfing, okay, you can get a loss. That means you are facing a loss of vigor and fertility during inbreeding. That is your inbreeding depression. Okay, loss of vigor and fertility. As I told, why we will see inbreeding depression, that too in cross-mated crop plants, as I already explained in the previous classes. Okay, the loss of vigor and fertility is due to recessive genes which are masked by the dominant genes in the CP crops. Okay, in case of self-planted crop plants, as I told, they are already exposed to the environment and they lost in the evolution if it is deleterious in nature. But in case of cross-planted crop plants, since they love the heterozygosity, your recessive allele will be masked by the dominant allele. They will be not expressed or they will not exposed to the environment. But what about inbreeding? You are doing it intentionally. The crops will never entertain inbreeding. Okay, inbreeding is your process. Okay, as a human intervention, you are doing inbreeding so that those recessive allele will get exposed to the environment. That will cause your inbreeding depression. विनायक, अरे यू लुक लाइक गोल्डन स्टार गणेश. अरे भाई ये सारे छोड़ दो. ठीक है, आप भी एक मैसेज डाल दो मुझे. ठीक है, मैं पर्सनली बात करेंगे. कर्नाटक से क्या विनायक? Sinchan deleterious recessive gene exposed and called inbreeding depressions. Yes, and the luxuriance. Actually, it is not true heterosis. Okay, your luxuriance is just, okay, as Kavita told, it is an high vegetative growth. It is false heterosis. ठीक है? It is not a true heterosis. It is just okay luxury in the growth. They will give some good vegetative growth, the good vegetation, okay, the good crop growth period. Your polyploidy, that is luxuriance. That is not true heterosis. Okay. By power negative and positive heterosis. What exactly the positive and negative heterosis? That's what. Okay. Your positive heterosis will be like for yield, for example. Okay. For example, yield. What you want? You want in the positive direction. Your yield should be always high. Okay, that is your positive heterosis. But in case of some pulses, for example, in red gram or any pulses, what you will expect? You will expect the number of growth period will be very less. The crop duration, okay, the number of days to flowering, you will expect some less number of days. Okay, that is also heterosis again. If you are minimizing the crop growth period, okay, we will also consider as a heterosis, but in case in a negative way. Okay, that is your negative heterosis. Okay, both are heterosis only. And the thing, one more thing I want to add here, your hybrid vigor and inbreeding depression are the two faces of the same coin. Okay, wherever you will try to improve the heterosis, okay, there is an high chance of inbreeding depression. The same faces of the two faces of same coin. Okay, clear? This is the difference. Result from outbreeding or crossing unrelated parents. Inbreeding is inbreeding due to related species. Heterozygosity will increase, here decrease. Common in cross-protected croplands. Here also it is common in cross-protected croplands. Increase in vigor and yield, decrease in vigor and yield. Measurement over mid-parent, better parent, standard variety. It is decline from F1 to F2. Okay, how your inbreeding is causing the loss of vigor from F1 to F2. If you are going from F1 to F2, there is a loss of vigor due to homozygosity. That how much decline, we'll measure it as an inbreeding depression. Average heterosis, heterobeltosis, and we will see what exactly it is. Cannot be easily fixed, lost in next generation. Okay, your hybrid vigor, and it is harmful gene become fixed in case of your inbreeding depression. They will fix in the homozygous condition. Dominant genes mask recessive gene. Here recessive harmful genes expressed. Superiority over parents, reduction in compared to F1. Seen in F1 generation, inbreeding depression can be seen in F2 and onwards. Okay, this is difference between heterosis and inbreeding depression. Next, in different degree of inbreeding depression. High inbreeding depression seen in alfalfa and carrot. Moderate will be in maize and sorghum and pearl millet. Okay. Low inbreeding depression will be onion, cucurbits, rye and sunflower. These are the things you should remember. Maybe they can ask. Okay, they can give in the match the following, and they can ask which are the crops which are having high inbreeding depression, less inbreeding depression. Okay, the moderate, no inbreeding depression in your self-planted crop plants. Okay, your maize and sorghum and pearl millet are having moderate inbreeding depression. This is very important now. Okay, your dominance hypothesis. Okay, over dominance hypothesis, and another one is epistatic hypothesis. These three I will explain the genetic basis of heterosis. First one is given by Davenport. Okay, later elaborated by Bruce, Kibley and Pello. What exactly it will tell? Dominance hypothesis. As I already explained in the PYQ also. Your dominance hypothesis, the heterosis is due to the masking effects of recessive allele by the dominant gene. Okay, whatever the recessive genes are there which are masked by dominant gene, so that you can expect heterosis. Okay, for example, it is the number of number of dominant gene is directly proportional to the heterosis. Whether your dominant gene is in homozygous condition and heterozygous condition, it doesn't matter. Both will give heterosis. This what this theory will tell. Okay, here your small a will be masked by capital A. Here there is no small a, so both will be effective. The number of dominant gene will be directly proportional to heterosis. You can see here inbreed 1, small a is having 1 unit. Okay, your capital B will give 2 units. Like that. Small c again 1 unit, capital D 2 units, small e 1 unit. So that there will be a total 7 units. In the same case, in inbreed 2, okay, capital A 2 units, BB 1 unit, capital C and capital E will give 2 units each. So that you will get an 8 units. Okay. What about in F1? F1 is heterozygous between 2 inbreeds. So capital A small a will give the same value as capital A unit, 2 units. All will give 2 units, total 10 units. This is the heterosis. Okay. If all are in homozygous condition, capital A, capital B, capital C, capital D, capital E. In this condition also, your value will be 10 units only. All will give 2 units. Okay, whether maybe they are in heterozygous condition, heterozygous dominant or homozygous dominant, they will give the same value. This what exactly your dominance hypothesis will tell. Okay, the heterosis is due to masking effect of your dominant gene. Okay, the masking effect of recessive allele by the dominant allele. Okay, this is what exactly your dominance hypothesis will tell. The number of dominant genes is directly proportional to heterosis. But in case of your over-dominance hypothesis, which is proposed by Shull, Fisher, later East and others, you can see capital A will give 2 units. Capital B will give 2 units. Small c 1 units, like that. When you will cross, okay, what you will get? Okay, your F1 generation, you will get heterozygous condition. Your heterozygous condition will give 3 units. Okay, here the thing is number of heterozygous loci directly proportional to heterosis. Number of heterozygous loci. Okay, both your capital A small a is superior to both capital A and small a small. It is superior to both. Your heterosis is due to heterozygosity itself. It is not due to masking effect of that particular recessive allele by the dominant allele. It's not like that. It is heterozygosity itself. You can see your capital A is giving 3 unit. It is 2, it is 1. But in case of your dominance hypothesis, okay, your both capital A small a are superior over small a. Both are 2 units, here 1 unit. समझ गया ना? Can inbreeding depression occurs in self-pollinated crop if the hybrid F1 to F2 is going? अरे सिंचन मैं already बताया था. In self-pollinated crop plants, okay, whatever the recessive genes are there, they are already exposed in the evolution and they are removed from the population. Okay, if any recessive genes are there, maybe you can see in F1 after in F2. Okay, those are not harmful. There is no inbreeding, so that there is no loss of vigor and fertility. There is no inbreeding depression in case of self-pollinated crop plants. फिर से पूछ रहा है तो ये समझ गए ना, between dominance hypothesis, over dominance hypothesis. In case of dominance hypothesis, the heterosis is due to masking effect of recessive allele by the dominant allele. Okay, but in case of over dominance hypothesis, okay, your heterosis is due to heterozygosity itself. बताओ, comment में बताओ. So there is one problem here in case of dominance hypothesis. Okay, as the principle, as the definition tells. Okay, so failure of, there is some objections now. Failure to isolate inbreeds as bigger as F1. Okay, dominance hypothesis telling that, okay, your capital A similar as capital A small a, then you should isolate the inbreed like this: capital A, capital A, capital B, capital B, capital C, small d, E, which is very superior. Okay, it is same as bigger as our F1, that is capital A, small b, capital B, small b, capital E, capital C, small c, capital D, small d, capital E, small e. It is same vigor as F1. You should isolate this inbreed. This is inbreed, right? As F1, effective as F1. But failure to isolate the inbreed like this. Why? This objection is explained by Jones. Okay, the linked gene hypothesis. What he will tell that the all dominant genes are not, they are linked with some recessive genes. The all dominant genes are not linked with each other. Okay, maybe capital A, there are some inbreeds, they are like capital A, small b, capital C, small d, capital E, capital D. They are linking like this, so that you can't isolate the inbreed as same as your F1. Okay, the same as your F1. You can't expect this particular genetic constitution of inbreed. It is very difficult since your dominant genes are linked with recessive genes. Okay, it is explained by Jones. We will call it as a linked gene hypothesis. Okay, the symmetrical distribution in the F2. What is exactly symmetrical distribution in the F2? Yes, if this particular dominance hypothesis, as it told, okay, we will get an capital A, small a, 1:2:1. Okay, these two are what? These two are giving the same heterosis, their same similar performance, as I told, as in case of dominance hypothesis. So this should be 3:1. If it is 3:1, then 3:1. Okay, what it is? If it is like graph, then it should be what it is? Skewed distribution. But what you will experience in the F2? You will experience symmetrical distribution in the F1. But according to this hypothesis, you should expect this schematic representation. It is also explained by Collins. Okay, this particular explanation also explained by Collins. Okay, that particular objection. And magnitude of heterosis. Okay, the magnitude of heterosis in case of dominance hypothesis. Okay, as I told, it is due to this particular condition. Okay, it should not like over dominance hypothesis. Okay, it is also one of the objection. Progressive heterosis in tetraploids. In tetraploids, again, there will be no number of numerous different combination of genomes. Okay, you should get some progressive heterosis. But again, according to this hypothesis, It will not explain that progressive tetracycline. And faster inbreeding repression in case of tetraploids. Again, this is the one of the objection. Okay, in case of your in the tetraploids, okay, since it contains the different genomes, it should not like faster inbreeding depression. There is a different genomes, right? In case of tetraploids, it should show some less inbreeding depression, but it will show the faster inbreeding depression. There is no again explanation for this particular objection. Okay, these two are explained. Okay, failure to isolate inbreed as explained by Jones in case of linked gene hypothesis, and symmetrical distribution F2, that is also explained by Collins. Okay, this is about your dominance hypothesis. Then over dominance hypothesis, it is heterozygosity itself. Okay, your over dominance hypothesis. Then epistasis. Epistasis is nothing but the heterosis is due to the interaction between the loci. Interaction between two or more loci. It is given by Gowan. Okay, Gowan. Now at present, okay, if I ask you a question that which is the most accepted hypothesis, genetic basis of the heterosis, okay, it is your dominance hypothesis, not over dominance. The dominance hypothesis is accepted genetic basis, but at present molecular innovations, after we know each and everything, the different principles, okay, now it is that we are concluding that, okay, your heterosis is due to combination of dominance, over-dominance and epistatic interaction. Okay, it is like a combination of all these three. If anyone asks you, you should be able to answer like this. Then there is a formula for heterosis. Okay, the Falconer gave mathematical expression for mid-parent heterosis. Okay, whatever you will get the heterosis in the F1, okay, it should directly summation of dy square. Okay, what is d? The degree of dominance, the deviation from the F1, how much it is deviating, that is your degree of dominance. Okay, it should be always maximum. Your degree of dominance should be maximum, so that you can expect the more heterosis in the F1. And y square, y is nothing but the difference between the gene frequency between the two parents. Okay, you are crossing between two parents, right? The difference in the gene frequency. Okay, if the sum of, if the divergence between the two parents, we can also tell like this: the divergence or the gene frequency between the two parents, that also it should be maximum. 1 is maximum, the y value maximum will be 1. If it is having 1, then your heterosis should be maximum. If it is 0, there is no gene frequency, the difference in gene frequency between two parents, then if it is 0, then your heterosis will be 0. Okay, there should be a degree of dominance, then there should be a complete. If it is 1, then it is completely difference between the two parents in the terms of gene frequency. Okay, and summation, why we will do the summation? We are not calculating for the single loci, it is summation for the over all the loci in the genome. Okay, this is about the heterosis. Okay, the fixation of heterosis you can do with the asexual or vegetative reproduction. If you fix the heterosis once, then you can go for vegetative reproduction so that you can maintain. And apomixis, as we already discussed it in the yesterday's class, how we can fix the heterosis through the apomixis. Then balanced little system, this is also we have discussed in the PYQ. Balanced little system is nothing but your homozygotes will die. Okay, your homozygotes will die, only heterozygotes will maintain in the population, so that you can fix the heterosis. And polyploidy, as I told, okay, in allopolyploidy. You can fix the heterosis. Okay, how? As I told, your allopolyploidy is itself is the crossing between two different species. Okay, whatever which is maintaining, it is heterosis itself. Okay, the difference between two different genome or three different genome. For example, wheat, capital A, capital A, capital B, capital B, capital D, capital D. It is just as your the heterosis. Okay, allopolyploidy. Yes, good. Then estimation of heterosis, average, the mid-parent heterosis, that is nothing but your F1 minus mid-parent divided by mid-parent into 100. Okay, heterobeltasis, F1 better parent into 100. Your useful or economic heterosis, when you will call it as a useful or economic? When over the check, the national check. F1 minus commercial check into 100. Standard heterosis. Okay, F1 minus standard hybrid. Okay, you are considering some of one of the any hybrid as a standard hybrid. Okay, over that particular standard hybrid into 100. Sometimes we will merge these two, standard heterosis and economic heterosis. Okay, used for check variety. Okay. Then types of different classes. Single classes, simple inbred classes, I1 into I2. Okay, double class, I1 into I2, I3 into I4. This is your double class when you are using 4 inbreds. Okay, three-way cross, the 3 inbreds. Modified single cross. This is nothing but you are crossing your first inbred with any sister lines of that particular inbred. Okay, then you will crossing with the second inbred. That is your modified single. Wherever you are having the modified word, okay, that is you are crossing with any sister lines first. Then double modified single cross. That means you are crossing your particular inbred with both inbred with the sister inbreds. Okay, double modified. And what about modified three-way cross? Modified three-way cross is nothing but I1 into I2, and again you are using I2 with the any sister lines of other inbred. Okay, in three-way cross you are using one sister line. That is your modified. And simple volatile hybrid. If you are crossing any two variety and making an hybrid, that is volatile hybrid. And top cross, you are crossing any variety with the inbred. Is it to estimate GCA? Means any variety with a tester. That is your top cross. Okay, this is all about the different crosses. Maybe in PYQ they have asked about it already. Okay, you should be very careful. Single cross, double cross, three-way cross, modified single cross, double modified. Okay. Then how you will develop, how you will produce the hybrid seed? First, before that, okay, you should know the, first you will develop an inbred. Okay, first you choose the inbred, or you can isolate the inbreds from the population. Okay, the first cycle inbreds are nothing but from the OPVs, directly from the heterogeneous population. We will isolate some hybrids. We will call them as a first cycle inbreds. Then what about second, third and fourth cycle inbreds? From hybrids or already developed inbreds. You are having the set of already developed inbreds. From those set of inbreds, you are trying to isolate some the new inbreds, or from the hybrids. Those are your second cycle inbreds. Okay, what you will do? First you will isolate an inbred. You will go to the field and you will select some superior plants and you will do the inbreeding. Then you are having some inbreeding, the set of inbreds, the pool of inbreds. Then what you will do? First, you will start with the phenotypic evolution. You are having some 100, 200 inbreds. First, you will go to the field and you will see for the phenotypic. Okay, whether you are using that inbreds as a male parents, whether you are using those inbreds as a female parents. Based on that, you will get shortlisted. You will shortlist some inbreds. If you are using as a male parents, then you should see for the plant height. It should be maximum. It should be high plant height. Okay, since the pollens will be in the upper end. And you will look for tassel length, the pollen production capacity. If it is female parent, you will look for cob length. You will look for the plant height should be small. Okay, this is the phenotypic evaluation if you are using as a male parent or female parent. Okay, then you are shortlisted some 100 or 50 lines. Then you will go for top cross test. That is nothing but you will cross with the OPV for testing the GCA. Then out of 100 lines, okay, you will test for GCA, and if whichever inbreds are giving good progenies, okay, you will select some 50 inbreds, for example 20 inbreds. Okay, after GCA, you will end up with 20 GCA, 20 inbred lines. Then those 20 inbred lines, you will go for all possible crosses. That means 20 means n of n minus 1 by 2. Okay, 20 minus 19 by 2, 10, total 190 crosses. You will go for single, that we will call it as a single cross test. So out of those 20, you will go for all possible single crosses. You will end up with 190 cross. Out of 190 cross, you will evaluate the F1s now. Okay, you will make a 190 cross now. So out of 190, you will get some superior 10, maybe 10 superior crosses. You will evaluate those 10 superior crosses. Okay, out of that you will release any 1 or 2 as an hybrid seed. This is the chain. In case of private companies, that will be like the continuous process. Okay, there will be multiple chains, multiple seed chain, multiple pipeline will be going on simultaneously. Okay, it will be like that. It is not that they will start with inbreed and they will wait for 4 or 5 years, then they will release hybrid. It will be continuous process. For every year, they will try to release. For every year, they are having all 4 steps are running in their back end. So they can release year by year based on the current scenario. Okay, the one other at the end, prediction of double cross performance, they will definitely ask in your exams sometimes. Okay, how we can predict the double cross, as we are already discussed about it. Okay, if you want to predict this particular double cross, okay, we will get F1, then you will cross, then you will get F1. Okay, if you want to particularly predict this F1 performance, then you should go for average performance of non-parental crosses. Okay, what about non-parental crosses? Non-parental, it is already parental cross. What about non-parental cross? That is your A into C, A into D, B into C, B into D. Okay, you should take an average of this particular non-parental crosses. Okay, so that you can, the average value of this particular cross will give you the performance, or you can predict the double cross hybrid. But only progeny selection leads to variety, sir. Right, variety is nothing but your hybrid is also in variety. Okay, don't think that the homozygous material is only the variety. Okay, your hybrid is also variety. Okay, your pure line selection is also in a variety. Pedigree selection, the end product of pedigree selection also variety. Variety, when you will consider variety, when you will push for the trial and the release, those are your varieties. Okay, don't think that variety is different, hybrid is different. Okay, whenever we will call, whenever we release hybrid, okay, we will call it as a hybrid variety. Okay, the simple thing. Production of hybrid seed, अरे वही बताया था ना मैं अभी. Okay, production of hybrid seeds is nothing but how we can produce hybrid. We can go for and emasculation. Okay, and emasculation, where and emasculation is advantage, where you can exploit. Okay, by single and emasculation, you will get more number of seeds. For example, in chili, tomato. Okay, or single emasculation, you will get the more number of seeds. There it is feasible. Otherwise, you should go for male sterility, self incompatibility. Okay, CHA, chemical hybridizing agents, and your transgenic male sterility. Okay, by using these different methods, you can produce the hybrid seeds. Okay, batao. Improvement, how you can improve the inputs by year, okay, you should go for the improvement of inputs. Otherwise, the same input, if you are using this the pipeline, then it is waste. Okay, as a farmer, everything they will expect the more, the yield should be very much more. So your input should be timely, there should be improvement. By using pedigree selection, backcross method, these are the methods you already know. Okay, the pedigree selection, by using pedigree selection, you can improve by crossing two different inbreds. And backcross method, by using, you can exploit, you can add some advantageous genes. For example, disease resistance, anything for the improvement for the inbreds, or any the novel traits you can upload to the old ones. Convergent improvement, just you are cross between any two inbreds which are having, the both are having some good character, you can just converge them into single inbred. Okay, it is given by Riche and Stadler. In PYQ, last PYQ, I think I have explained about it. Gametic selection. Okay, gamete selection. What we will do? This is your inbred, the group of inbreds. First, you will cross with OPV. Okay, then you will, what you will do? You will store some seeds. Okay, you will grow the progenies. First, you will grow the progenies of this particular OPV. You will cross with tester. Okay, you will cross with tester. Whichever progenies are giving good yield, okay, you will select those things and you will go for intercross among themselves. Okay, that means you are selecting for gamete. Okay, you are selecting from the OPV. You are choosing some good pollens. Okay, and you are accumulating in the inbreed, so that then you will intercross among them. So whichever giving the best with the tester, you will intermate among themselves. Okay, where your gametes are superior, okay, over the zygotes, okay, there you will use this particular gametic selection. Then somatic hybridization. These are some genetic engineering techniques. Okay, by using these techniques also you can improve the inbreed lines. Okay. Yes, Jeevan. Abhi sir, बताओ. जीवन बताओ, कुछ डाउट है क्या? अभी हम हाइब्रिड ब्रीडिंग पढ़ा रहा है, ठीक है? हाइब्रिड के बारे में कुछ डाउट्स है क्या? अभी बताओ, कोई बताओ, कुछ डाउट्स है क्या? अभी हाइब्रिड तो हो गया खत्म. What are the merits? Hybrids are higher yield than synthetics composites. We will see what exactly synthetics and composites now. More uniform and attractive. Can be developed in both self and cross-planted croplands. Same hybrid can be reproduced with the same genotypes. Okay, if you are already having inbreeds, you know the inbreeds and you know the genetic constitution of particular inbreed, so you can easily reconstitute that particular inbreed. Okay, that is these are the merits. Okay, highly homogeneous. Okay, highly homogeneous. They will give good yield. But what are the demerits of hybrids? Farmer must buy the new seeds every year. Okay, if you use the same seeds, the hybrid seeds in the next generation, there will be segregation. Okay, then he will see some are giving good yield, some are giving low yield. It will be very loss to the farmer. Okay, so he should purchase each and every year for the hybrid seed. For means if you want to buy, if you want to grow an hybrid, then you should purchase the seeds each and every year. Costlier seed than other varieties. Your hybrids obviously costly since you are giving some efforts, right? You are making yarn demasculation, you are producing hybrid seeds. It is costlier method. Then need more inputs for hybrids. And hybrid seed production is more skill and technical management. Okay? Plum room thin. Okay, no sir, all clear. जीवन आप मैंगो खा सकते हैं वो कबोर्ड में, ठीक है? So to avoid these demerits, ठीक है, we can go for synthetics and composites. ठीक है? What exactly the synthetics and composites? What is the base population for this? Okay, as I told, your recurrent selection is the base material for synthetic variety. Okay. Viewers तो 30 आ गया, सारे चला गया क्या? मैं तो सारे ग्रेड का कोर्स इधर ही खत्म कर दिया, ठीक है? There are some 5-6 slides, okay? अरे कुछ जोश दिखा दो, क्या हो गया सबको? सारे चला गया क्या? जोश इज आई सर, ठीक है, ठीक है, बीट bounce, okay. What about synthetic variety? First, what you will do? You will do the isolation of inbreeds by recurrent selection only. Then, okay, you will isolate the inbreeds. Evaluation for GCA. In the recurrent selection itself, you will already evaluate. But if you want to, you are doing the separate process for synthetic variety, then you will go for evaluation of GCA. Then whichever the inbreeds which are having good GCA, okay, what you will do? You will intermate of the selected inbreeds, and you will take the equal amount of seeds from the intermating, and you will bulk. Okay, then you will give to the farmers. Simple. In this particular thing, okay, you will test for GCA. The inbreeds which are having good GCA, you will make an intermate among themselves. Okay, then you will give those F1 seeds or S1 or Sync 2 seeds, not Sync 1, you will not give. Then you will go for one more generation, open pollination. Then Sync 2 seeds you will give to the farmers. Okay, what are the merits here? It exploits heterogeneity in crops where hybrid seed production is very difficult. Okay, where you can't cross, okay, planted crosses. Okay, where there is an open pollination, for example, bajra, sorghum. Okay, there it's very difficult to make a hybrid seed production. There you can use this particular synthetic variety. Seed is cheaper than hybrid. Okay, you are not going for any particular the different technology here. Broad genetic base, since it is heterogeneous material, heterogeneous, so it is better adaptability, and farmers can save the seeds for 4 to 5 years. Okay, since it is a mixture of different intermating selected seeds, so it is already heterogeneous, then there is no fear for segregation. Okay, it is already segregating material, so it is maintain the yield for certain generation. So you can go up to 4 to 5 years. more stable yield under the fluctuating environment since it is broad genetic base. Okay, broad genetic is nothing but heterogeneous material. Okay, it is a mixture of different genetic constitution. So it will maintain some stable yield. Factors determining the yield of synthetic variety. Okay, decline in heterozygosity in sync 2. What are the factors which will determine the yield? Okay, every year, okay, after sync 2, your heterozygosity will decline. But how much decline occurs? It depends on the number of parental inbred lines and genetic differences among those line. Okay, and decline in the synthetic performance, sync 2 performance. The formula is given by Sewell, right? The sync 2 performance is sync 1 minus sync 1 minus sync 0 divided by n. Okay, this is the formula of sync 2 performance. Okay, you can expect that particular synthetic 2 generation performance. Okay, your sync 2 is nothing but performance after one generation of random mating. Your sync 1 is average performance of all F1 crosses. Sync 0 is average performance of parental lines. Your sync 0 is parental lines, okay, the starting material. Sync 1 is after your intermating F1. And sync 2 is performance after one random mating. So your sync 2 seeds only will go to the farmers. You will go for one random mating, then you will go for distribution of seeds. Okay, but you should know the sync 2 performance. Okay, you can easily calculate. Okay. The thing is, how you can improve the sync 2 performance? How you can improve the sync 2 performance? You can see. One, you should improve the sync 1 performance. If it is your sync 1 performance will increase, automatically your sync 2 performance will increase. But how to increase this particular sync 2 performance, sync 1 performance? Okay, you should have the sync 0, they are having some good performance, the original material, and the number of inbred lines you can see here. Okay, the number of inbred lines are directly proportional to the sync 2 performance. If you increase this n value, the number of inbred lines, okay, if you are increasing this, so that you can decrease this particular value. If you are decreasing this particular value, okay, if it is minimizing, obviously your sync 1 will improve. So your sync 2 will improve. So your n is directly proportional to the performance, sync 2 performance. That too till some number of inbreds, okay, up to 12 inbreds. Okay, if you are increasing till the 12 inbreds, so you can improve the sync 2 performance. After 12 inbreds, if you are involving the more number of inbreds, then again your sync 2 performance will decrease. Okay, so your n should be increased. You should increase the number till some point. Okay, and also the original sync 0 performance of parental lines should be maximum. If it is maximum, okay, then it will reduce this particular value, again it will increase. Okay, your sync 0 performance should be very high and the number of inbred lines should be maximum. Okay, this is how we can increase the sync 2 performance. Next is composite variety. In cross-planted crops, okay, it is nothing but mixture of seeds from the several similar genotypes that are grown together, maintained by open pollination from generation to generation. It is just mixing and you are growing and you are leaving for open pollination. Okay, there is no testing for GCA, nothing. Selection for base material, then intermating selected genotypes, then evaluation of crosses, then mixing of seeds. Just you are selecting some good genotypes, you are making intercrosses, mixing, and you are giving to the farmers. Again, stable yield under different environmental condition. Cheaper seeds, farmer can reuse the seeds. Okay, and it is also a broad genetic base. The one demerit in this particular composite variety that you cannot be exactly reconstituted. Okay, since you are not tested for GCA, okay, and you are not started with particular inbreds which are having good GCA. You don't know from which the base material you have started this particular program. It is very difficult to reconstitute the particular material. Okay? This is about your composite variety. Next, breeding methods for vegetatively propagated crops. Very simple. ज़्यादा सोचने की जरूरत भी नहीं है इधर. ठीक है? Some crops are having essentially propagated, like where there is a node seed involvement, sugarcane, potato, sweet potato, banana. Okay, these are some different examples. Sexual segregation and recombination occur in case of sexually propagated crop plants. So there will be a new gene combination and variation so that you can go for selection. But in case of asexual, there is no recombination. Offsprings are very identical. There is no genetic variation. Okay, it is very difficult to apply any breeding methods. So what we will do, there are some techniques in case of asexual reproduced plants. First, before that, you should know the different characteristics. These are mostly perennial. Okay, many shows reducing flower and seed set. There is no flowering sometimes. Cross-noted mostly, highly heterozygous. So that's why they will show high inbreeding depression, severe inbreeding depression. Okay, and many are polyploids, often interspecific hybrids. Sometimes each variety is actually a clone. Whatever we will develop in this particular group of plants, okay, finally it is a clone. Okay. Clone is a group of plants produced from a single plant through the asexual reproduction. The one major advantage here, you can fix the heterosis, okay, in clonal crops. Once you develop any hybrid, then you can easily maintain those crops, those clones. What are the characteristics? Identical genotype, no genetic variation, immortality. Immortality means that it will be constant genetic constitution. It will not change over generation, but severe inbreeding depression. But how we can create some genetic variation? What are the ways? What are the origin of that particular genetic variation? You can see through the bud mutation, somatic mutations, okay, in the buds, mechanical mixture, and occasional sexual reproduction. In occasional, there will be some sexual reproduction. Okay, these are the origin of variation in case of your clones. And there is one word, that is clonal degradation. Yes, your clone is immortal, but sometimes it will degrade due to any virus, infection, okay, or mutation, the minor micro mutation which leads to the clonal degradation. Okay, this is about your clones. If you compare the clone, pure line and inbreed, mode of pollination, cross-mated as similar as your inbreed. The thing what you should target here is genetic makeup within the entity is heterozygous. Okay, but here in case of inbreed, it is almost homozygous. Okay, and again it is heterozygous and homogeneous. Clone will be heterozygous and homogeneous. Your inbreds are almost heterozygous and homozygous and homogeneous, your inbreds. But clones are heterozygous at individual level, and population level, again they are same, heterogeneous. Okay, obtained through asexual reproduction from the one plant. But here there is a selfing, again it is selfing or inbreeding. Okay, these are the things you should remember. What are the methods? Okay, a single outstanding plant can become a new variety in case of your clonal reproduction. Okay, two main phases in clonal crop breeding. You should create the genetic variation first. How you can create the genetic variation? Through natural mutation or hybridization. These are the two main things. If you want to create any variation in clonal crops, one, it is your mutation. Okay, you should go for mutation breeding in case of clonal crops. Otherwise, you have to start from hybridization. Okay, you should take some artificial hybridization. You should involve and you should make some starting hybridization program so that you can create some variation. After that, whatever it may be, whatever you are doing, mutation or hybridization, okay, any breeding method, any genetic engineering approaches, at the end of the program, at the end of the day, you should go for clonal selection. Okay, after you are introducing any mutation, after you are doing any hybridization, okay, you will just cross. Then at the last, you should start with a clonal selection. Okay, you will select some best clones after your mutation. Then you will go for that clones in multi-location trial. Then you will go for further process. Okay, whatever it may be, at the end of the day, we will go for clonal selection. True to type, yes. Kavita, chimera, sir. Chimera, वही बता रहा है मैं. Chimera, chimera. You will see now, chimera. Can see here. What is the problem with the chimera? Okay, the mono layer of cell, your L1, you can see, if it is shoot tip, you can take, this is your shoot tip. Okay, there are 3 layers always. Okay, L1, L2 is most inner layer, L3, where you can see only L1 in epidermis, but L2 in gametes or in some tissues. But in case of inner tissues, okay, in root or vascular tissue, you can also see the L3. Okay, you can see this is L1, L2, L3. Yeah, okay. L1, L2, L3. It is, for example, shoot tip initial. Okay, there are 2 types of chimera, sectorial and periclinal. Okay, what about sectorial? You can see this is one layer, complete layer, but half of the layer is having one of the genotype and the remaining portion will be other genotype. That is your sectorial chimera. Okay, you can see it is capital A and it is capital A small a, other sector. Okay, it is capital A, it should be capital A small a, the other sector in the same layer. That is your sectorial chimera. What about periclinal chimera? The complete layer should be different genotype, and another layer, the other layer should be different. But in case of sectorial, within this single layer, there is a 2 different genotype. Okay, it is due to mutation. Okay, it is obviously due to mutation. Okay, this is the chimera. Okay, this is we will call bud mutation. After this shoot tip matured, okay, some of the part will have some other genotype, some of the part will have having other genotype. We can separate that particular genotype which is having some other genotype and we can maintain as a clone. That is a bud mutation. We can exploit that bud mutation. Generally, most of the clonal crops mutant will become periclinal. First, it should be start with sectorial, then it will completely periclinal. Okay, that's what here written here. Generally, most of the clonal crops mutant will become periclinal even if there were mericlinal initially. Okay, mericlinal are nothing but your sectorial. Okay, sectorial are nothing but your mericlinal. This is about chimeric problem, and this is about again the courses. Okay, I will not mark it now, this particular thing. After listening this particular class from past 5 hours, now you can just imagine how much content we are giving through the courses. Okay, you can just get an idea. You will get an idea now. Okay, what are the information we are giving, how we are guiding the so many students now. Okay, again, definitely they are getting some benefits. Okay, these are all about our courses. Okay, you can just go to our website and you can just explore the content. Okay, this is all about today's class. Yes. Okay, regarding the plant breeding part 2. Okay, now I have completed till the breeding method of self, cross and asexually reproductive plants. Now we are left with stress breeding. Okay, with the quick revision we will do on stress breeding. Then after that, some special breeding methods, genetic engineering, transgenics, then markers, if yes, and IPR. We will cover each and everything in part 3. I will try to cover complete plant breeding. Okay, if you guys want part 3, plant part ज़्यादा mix हो रहा है मेरा पता नहीं. If you guys want part 3, as I already told, we need comments in both this particular video. Okay, and also you should share this video as much as possible, and also just comment on that video which it is in the description box regarding the new launch course video. Okay, you take our name or plant science batch name or my name or anyone's name, just give a comment so that everyone should know. Okay, everyone should get benefit from our platform. Okay, it's not about they should purchase the courses, it's about the information it should be passed through your juniors and to the all our country, so that everyone should get benefit from our agriculture community. That's what our dream is. Okay, clear? Any doubts in today's session? Kavita, Nemani, Madhu, batao, any doubts, anything to ask? Only 30 students remaining. अरे गाइस, उतना बोर भी नहीं था ना आज. 5 घंटे हो गया. उतना बोर भी नहीं था शायद. Can anyone tell what's your genuine feedback for these sessions? You can share, no problem, genuinely. What is where we want to improve? Where I can improve? You guys can share. I will take it positively. मस्त क्लास था सर, मजा आ गया. ठीक है, वेरी मच इंफॉर्मेटिव. अरे जेन्युइन फीडबैक तो दे दो थोड़ा. अरे कुछ नेगेटिव बताओ. कुछ नेगेटिव बताओ. मैं लास्ट सेशन में कुछ ब्रेक लिया था, अभी आज तो कुछ ब्रेक भी नहीं है, कुछ नहीं है. ठीक है. मेरे मतलब करना पूरा दर्द हो रहा है, पूरा बॉडी. कुछ नेगेटिव बताओ भाई. कोई बता सकता है क्या? सिंचन कहाँ चला गया? भाग गया क्या रिवीजन करने के लिए? हाँ, सिंचन कहाँ गया तू? Sir, revision is done completely in your classes. Yes, we are doing it, but someone should say some negative points so that we can improvise. Revision become easy with your session, sir. अरे वो तो ठीक है, मजा आएगा सबको. If you guys want to share anything, any feedback, I want some negative feedback. Increase the gift, sir. ठीक है. नेक्स्ट सेशन में मैं 3 से नेक्स्ट सेशन में 5 गिफ्ट. ठीक है, they will definitely give. I will come with 5 questions. Nikhil Chaudhary, I have prepared something special for dinner. Please come, sir. ठीक है, निखिल तो पता है ना, हमारा मेंटर था. ठीक है, वो वेट कर रहे हैं शायद खाने के लिए. Sir, when is the timing for class start? This will be going to be live every day till JRF 2026. अरे भाई, मैं हमारा YouTube चैनल में सारे लाइव हो रहा है, मतलब डेली एक तो एक रहेगा. आप आराम से कवर कर सकते हैं, रिवीजन कर सकते हैं सब कुछ. ठीक है, कोई नहीं, मतलब नेगेटिव फीडबैक तो नहीं दे रहा है मुझे. ठीक है, अच्छा लगा. Then we will meet in part 3. Okay, till then, good night each and everyone. As I told, if you guys need part 3, I am very serious about it. Okay, if you guys want part 3, that is very important. Again, as I told, stress breeding, they will definitely ask quality breeding. Okay, genetic engineering, markers, सब कुछ बचा हुआ है. If you guys want plant part 3, then you know what you should want to do here. Okay, regarding comments, likes and share, everything I want. Okay, you just see after the part 1, after it reached 4K, then only I am taking this particular part 2. Then it should also reach that much views, then I will come for part 3. Okay, and don't forget about the comments for that new launch course video. Okay, till then, good night, bye bye.